In the quiet moments before sleep finds us, our minds often wander to the invisible world that surrounds us, a world of microscopic life that has shaped human history in ways we're only beginning to understand. Tonight, we journey through one of the most remarkable stories in medicine, the discovery of antibiotics and the delicate balance we now find ourselves maintaining with the bacterial world. Consider this. In the year 2019, an estimated 1.27 million people died directly from infections that our antibiotics could no longer cure. These weren't deaths from rare or exotic diseases, but from bacterial infections that, just decades ago, would have been easily treatable. The number becomes even more sobering when we expand our view. Nearly 5 million deaths that year were associated with antimicrobial resistance in some way. 6 bacterial species were responsible for most of these tragedies: Escherichia coli, Staphylococcus aureus, Klebsiella pneumoniae, Streptococcus pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. Names that may sound unfamiliar now, but ones we'll come to know as we move through this story. The future projections paint an even more striking picture. Between 2025 and 2050, over 39 million people could lose their lives to antibiotic-resistant infections. By the middle of this century, deaths from antimicrobial resistance may rise by nearly 70% compared to 2022. The United Nations has warned that without intervention, drug-resistant infections could claim more than 10 million lives globally each year by 2050. A number that invites us to pause and reflect on the fragility of the medical advances we've come to take for granted. This scenario has been given a name that captures its gravity: the Post-Antibiotic Era. It's a term that describes a potential future where antibiotics become ineffective, where medicine might return to conditions resembling the time before these wonder drugs existed. In such a world, common infections could once again become deadly. A simple cut could lead to an untreatable infection. Surgeries that we now consider routine would carry unacceptable risks. Cancer treatments, bone marrow transplants, and organ transplants, all of which require suppressing the immune system, could become impossible to perform safely. The economic implications are equally profound. Projections suggest that antibiotic resistance could erase up to 3.5% of global GDP by 2050. In China alone, resistance could cause the loss of up to 1 million lives each year by mid-century. These aren't just statistics. They represent a fundamental challenge to modern medicine and the structure of our healthcare systems. Yet this is not a story of inevitable decline. It's a story about human ingenuity, about accidental discoveries and systematic research, about the intricate dance between innovation and evolution. It's about how we got here, how bacteria respond to our interventions, and what we might do to preserve these precious tools that have extended and saved countless lives. So settle in, let your breathing slow, and join us as we trace this remarkable journey. We'll begin at the dawn of the 20th century, with the first attempts to create drugs that could kill bacteria without harming the patient. We'll witness serendipitous discoveries in London laboratories and systematic production efforts during wartime. We'll explore the elegant mechanisms by which antibiotics work and the equally elegant ways bacteria have learned to resist them. And we'll end by looking toward the horizon, at the innovations and approaches that offer hope for maintaining our edge in this ancient evolutionary contest. The story of antibiotics is, in many ways, the story of modern medicine itself. Let's begin. Our story begins in the early years of the 20th century, in a time when infectious diseases claimed lives with a regularity that would seem unimaginable today. It was an era when a simple scratch could lead to blood poisoning, when pneumonia and syphilis ravaged populations, and when physicians had few tools beyond supportive care to offer their patients. Into this world stepped a German physician and scientist named Paul Ehrlich, whose systematic approach to finding treatments would forever change medicine. Ehrlich was fascinated by a curious observation. Certain synthetic dyes could selectively stain specific pathogens under a microscope without affecting human cells. If dyes could find and mark these organisms, he reasoned, perhaps chemicals could be designed to find and kill them as well, to act as what he called a magic bullet, or Zauberkugel in his native German. It was a revolutionary concept, a drug that could selectively destroy disease-causing microbes while leaving the patient unharmed. His interest in targeted drugs had begun in the 1890s when he experimented with methylene blue for treating malaria. Later, he studied trypan red against trypanosomes, the parasites causing African sleeping sickness. But his most ambitious work began after the discovery, in 1905, of Treponema pallidum, the spirochete bacterium responsible for syphilis. Ehrlich turned his attention to atoxyl, an arsenic compound that had shown some promise against trypanosomiasis, and began what would become a methodical, exhaustive search. Working with his colleague Sahachiro Hata at his laboratory, Ehrlich systematically synthesized and tested variation after variation of arsenic compounds, more than 1,000 different candidates in all. They tested each one in infected rabbits, documenting their effects with meticulous care. This wasn't the work of lucky accident, but of deliberate, systematic experimentation. Compound after compound failed, showed too much toxicity, or proved insufficiently effective. But Ehrlich persisted. Finally, compound number 606, arsphenamine, emerged as the magic bullet he had sought. Synthesized in 1907 by his colleague Alfred Bertheim, its antisyphilitic activity was discovered by Hata in 1909 during this systematic survey. Introduced to the medical world in 1910, it became known as salvarsan, and it represented the first effective treatment for syphilis, the first modern synthetic antimicrobial agent. For the first time in history, a man-made chemical compound could cure a bacterial infection. Salvarsan wasn't without its challenges. The drug had to be dissolved in several hundred milliliters of distilled, sterile water with minimal exposure to air, a complex preparation that made administration difficult. The minimum treatment period lasted 18 months and required 20 salvarsan injections along with 30 to 40 bismuth injections. Yet despite these difficulties, it worked. In 1914, a second-generation drug called neoarsphenamine, or neosalvarsan, was developed with improved characteristics, making treatment somewhat easier. Salvarsan remained the standard treatment for syphilis until the early 1940s, when penicillin would finally replace it. But Ehrlich's legacy extended far beyond salvarsan itself. He had established a paradigm, a systematic screening process that would guide pharmaceutical development for generations to come. His concept that drugs could be designed to target specific pathogens, his methodical approach of synthesizing and testing hundreds or thousands of variations, his belief that chemistry could solve medical problems. These ideas became the foundation of modern drug discovery. 2 decades later, another German scientist would follow in Ehrlich's footsteps, bringing the same systematic rigor to the search for antibacterial agents. Gerhard Domach was a pathologist and bacteriologist who, beginning in 1927, worked at the German industrial giant IG Farben. His task was to test chemical compounds for potential medical applications, continuing the tradition that Ehrlich had pioneered. In the early 1930s, Domach tested a novel compound that chemists Friedrich Miech and Joseph Clara had synthesized, a benzene derivative of an azo dye, with a sulfonamide group attached. They called it sulfonamidochrysoidin, though it would become better known by its trade name, Prontosil. When tested against Streptococcus pyogenes in laboratory animals, the compound showed remarkable antibacterial activity. The moment that would cement Prontosil's place in medical history came in 1935, under circumstances every parent dreads. Domach's daughter Hildegard injured herself and developed a streptococcal infection. As the infection worsened and threatened both her arm and her life, Domach made a desperate decision. He administered the experimental compound that had shown such promise in animals. The result was dramatic. His daughter recovered. Prontosil worked. But the story of recognition for this achievement carries its own dark chapter. Domach was awarded the 1939 Nobel Prize in Physiology or Medicine for discovering the antibacterial effects of Prontosil. However, the Nazi government, which had grown hostile to the Nobel institution, prohibited him from accepting the honor. When he attempted to accept anyway, he was arrested and imprisoned. Only after the fall of Nazi Germany in 1947 could he finally accept his Nobel diploma and deliver his lecture, though by then the monetary prize had expired according to the rules of the time. Prontosil marked the beginning of the sulfonamide era. Scientists soon discovered that the active component of the drug was actually sulfonilamide, a simpler compound that had been synthesized as early as 1908, though its antibacterial properties had gone unrecognized for nearly 3 decades. This revelation was crucial. Sulfonilamide was not patentable, as it had been previously synthesized, which meant it could be produced by any pharmaceutical company. The sulfa drugs, as they came to be known, spread rapidly through the medical world. These were the first widely used antibacterial drugs in history, predating the widespread clinical use of penicillin by nearly a decade. As the first and only effective broad-spectrum antibiotic available in those years, sulfa drugs saved tens of thousands of lives during the early years of World War II. Among the notable figures whose lives they preserved was Winston Churchill himself, who was cured of a potentially lethal pneumonia with a sulfa drug called sulfapyridine in late 1943. Today, sulfonamides remain on the World Health Organization's list of essential medicines, a testament to their enduring value. Now we must step back in time slightly, to September of 1928, to a laboratory at St. Mary's Hospital in London. Here, a Scottish bacteriologist named Alexander Fleming was conducting research on Staphylococcus aureus bacteria. The discovery he was about to make would become one of the most famous stories in the history of medicine, though, as we'll see, the story we often tell isn't quite the whole truth. The traditional narrative goes like this. Fleming returned from a 2-week holiday on September 3, 1928, and began sorting through petri dishes containing colonies of Staphylococcus. He noticed one dish had been contaminated with a patch of green mold, later identified as Penicillium notatum. Around this mold, something remarkable had occurred. The bacteria had been killed, creating a clear zone where the mold secretions had prevented the staphylococci from growing normally. Fleming identified that the mold was producing a substance capable of killing a wide range of harmful bacteria, and he named this substance penicillin. Fleming himself later claimed that he awoke just after dawn on September 28, 1928, and certainly didn't plan to revolutionize all medicine. This date, September 28th, has been enshrined in history as the day of the discovery. It's the date we celebrate, the moment we commemorate. But when we look more carefully at Fleming's own laboratory notebooks, a more complex picture emerges. The earliest recorded experimental mention of the mold and penicillin in Fleming's notes is dated October 30th, 1928, nearly 2 months after that supposedly fateful September morning. And this October entry doesn't describe a chance discovery of a contaminant. Instead, It documents a carefully constructed experiment using what's called the reverse sequence culturing method. The mold was cultured first, and then bacteria were deliberately added around it. This suggests something important about the nature of scientific discovery. Between that moment in early September when Fleming first noticed the contaminated dish and the October experiment carefully documented in his notebook, he had been working, isolating the mold, identifying it, characterizing its properties, determining the conditions under which it produced its antibacterial substance. The discovery wasn't a single moment of serendipity, but a process of careful, methodical scientific investigation. The serendipitous element was real. Fleming didn't intend for his cultures to become contaminated. But the discovery itself involved more deliberate inquiry than the popular narrative acknowledges. Fleming had to recognize that what he was seeing was unusual and important. He had to have the knowledge and curiosity to investigate further rather than simply discarding the contaminated dish. He had to do the work of isolation and characterization. Science, it turns out, favors the prepared mind, even when that mind is presented with an unexpected observation. This serendipitous yet systematic discovery marked a turning point in human history. Fleming had identified a substance produced by a common mold that could kill dangerous bacteria. But the path from that contaminated petri dish to the mass production of penicillin as a life-saving drug would take more than a decade and the efforts of many more scientists. That part of the story, though, belongs to our next chapter. For now, as we pause at the threshold of the 1930s, 3 paths to antibacterial drugs had been illuminated. Ehrlich had shown that systematic chemistry could create targeted antimicrobials. Domach had demonstrated that industrial research could bring these drugs to widespread use. And Fleming had revealed that nature itself had already been producing antibacterial substances. We needed only to find them and learn to harness them. The age of antibiotics had truly begun. As the 1930s gave way to a new decade, Fleming's discovery remained largely a laboratory curiosity. He had published his findings in 1929, but penicillin proved difficult to produce in meaningful quantities and unstable when isolated. It would take the outbreak of World War II and the convergence of scientific minds working under the pressure of wartime necessity to transform this promising observation into a medicine that would save millions of lives. In the late 1930s, at the University of Oxford, 2 researchers took up the challenge that Fleming had set aside. Howard Florey, an Australian pathologist, and Ernst Boris Chain, a German-born biochemist who had fled Nazi persecution, assembled a team to investigate promising antimicrobial substances. Among their candidates was Fleming's penicillin. Chain succeeded in purifying the compound in early 1940, a crucial breakthrough that had eluded Fleming himself. With purified material in hand, Florey and Chain could finally conduct proper tests. In May 1940, they reported penicillin's therapeutic value in mice infected with lethal doses of streptococci. The results were striking. Mice treated with penicillin survived, while untreated controls died. By early 1941, they had moved to testing the drug on human volunteers. The first formal patient treatment came in February 1941. A policeman suffering from a severe infection. Though the initial supplies ran out before the treatment could be completed and the patient ultimately died, the therapeutic effect during treatment was unmistakable. Penicillin worked in humans. The first civilian patient to be fully and successfully treated was a woman named Ann Miller. In March 1942, she lay near death at New Haven Hospital in Connecticut, suffering from blood poisoning following a miscarriage. Her temperature had spiked to nearly 107 degrees Fahrenheit. The few precious doses of penicillin available in the United States were brought to her bedside, and within hours of the first injection, her fever began to fall. Within days, she was recovering. And Miller would live for another 57 years, a living testament to the drug's power. For their collective achievement, Fleming's discovery and Florey and Chain's development of methods for isolation and production, all three men shared the 1945 Nobel Prize in Physiology or Medicine. The citation recognized them for the discovery of penicillin and its curative effect in various infectious diseases. It was a rare acknowledgement that scientific breakthroughs often require both the initial observation and the subsequent hard work of development and refinement. But recognition and small-scale success weren't enough. The world was at war, and soldiers were dying from infected wounds. What medicine needed was not carefully rationed doses produced in university laboratories, but industrial quantities. Enough penicillin to treat thousands, then hundreds of thousands, of patients. The challenge of mass production would prove nearly as difficult as the original discovery. Between 1940 and 1945, penicillin production underwent a revolutionary transformation, coordinated by the U.S. War Production Board. This wasn't the work of a single company or laboratory but a collaborative effort involving 21 companies, 5 academic groups, and multiple government agencies, including the U.S. Department of Agriculture. Scientists at the Northern Regional Research Laboratory pioneered what became known as deep fermentation techniques, growing the mold in large tanks of liquid medium rather than on surfaces. This innovation alone quadrupled yields within months. But improvements didn't stop there. Researchers at Cold Spring Harbor used X-rays to induce mutations in the mold strains, coaxing them to produce more penicillin. Yields increased from 250 to 500 units per milliliter. Scientists at the University of Wisconsin applied ultraviolet radiation to generate even more productive strains, nearly doubling output again. Between May and June of 1944, production rates increased more than 250-fold. The scale of the operation grew to match its ambition. By January 1945, American facilities were producing 4 million sterile packages of penicillin each month, using fermentation tanks with capacities reaching 10,000 gallons. Pfizer's Brooklyn facility alone operated 14 fermenters, each holding 7,000 gallons. After years of wartime restriction, penicillin was finally released for commercial public distribution in March 1945. What had begun as mold growing on a forgotten petri dish had become an industrial product that would reshape medicine. Yet penicillin wasn't the only antibiotic emerging during these transformative years. In September 1943, at Rutgers University in New Jersey, another crucial discovery was unfolding, one that would prove particularly important for a disease that had resisted all previous treatments. Selman Waxman, a soil microbiologist, had spent decades studying the microorganisms that lived in soil, particularly a group of bacteria called actinomycetes. Along with his graduate students Albert Schatz and Elizabeth Buggy, Waxman was conducting a systematic search through soil samples, looking for microbes that produced antimicrobial substances. Unlike Fleming's chance observation, this was planned, methodical research grounded in Waxman's deep understanding of soil microbiology. From the soil bacterium Streptomyces griseus, they isolated a compound they named streptomycin. What made this discovery particularly significant was its activity against Mycobacterium tuberculosis, the bacterium that causes tuberculosis. Tuberculosis was then a leading cause of death worldwide, and it was completely unaffected by penicillin. For the first time in history, physicians had a drug that could treat this ancient scourge. Streptomycin became known as the world's first broad-spectrum antibiotic, effective not only against tuberculosis but also against the bacteria causing plague, cholera, and typhoid fever, diseases that had terrorized humanity for millennia. Waxman received the Nobel Prize in 1952 for this achievement, and streptomycin joined penicillin in the rapidly expanding arsenal of antibacterial drugs. The late 1940s saw the pace of discovery accelerate. Benjamin Minge Duggar, an American plant physiologist working at Lederle Laboratories under the supervision of Yale Prugda Subburo, was continuing the systematic approach that had proven so successful. Rather than waiting for chance contamination, Duggar methodically cultured and screened actinomycetes from soil samples collected across the United States. In 1945, though some sources indicate the antibiotic was isolated in 1948, from a soil sample taken at Sanborn Field at the University of Missouri, Duggar identified an antibiotic produced by Streptomyces aureofaciens. The organism and its product were named for the molecule's intense golden yellow color, aureomycin, from aureus, meaning golden. The chemical name was chlortetracycline, and it represented the first member of what would become the tetracycline family of antibiotics. Chlortetracycline marked another milestone. Unlike penicillin and streptomycin, which came from specific microbial species, this discovery demonstrated that the actinomycetes in general were a rich source of antibacterial compounds. Today, soil actinomycetes are responsible for producing about 2/3 of all antibiotics used in hospitals and clinics. The wisdom of systematically exploring these organisms, rather than waiting for serendipitous discoveries, had been vindicated. Chlortetracycline itself remains on the World Health Organization's list of essential medicines. The final crucial innovation of this revolutionary period came in 1955, and it represented a conceptual leap as important as any of the discoveries themselves. Lloyd Conover, working at Pfizer, asked a question that hadn't been seriously considered before. Could existing antibiotics be chemically modified to create improved versions? Conover took chlortetracycline, aureomycin, and subjected it to a chemical process called hydrogenolysis, using hydrogen gas with a palladium on carbon catalyst in a mixture of methanol and dioxane. The reaction removed a chlorine atom from the molecule, producing what he called a deschloro product, tetracycline. When tested, this modified compound proved just as active as the original aureomycin. This was a revelation. For the first time, it had been demonstrated that antibiotics weren't fixed entities to be accepted exactly as nature produced them. They could serve as scaffolds, starting points for chemical modification. The naturally occurring compounds could be improved, simplified, or altered to create new medicines with potentially better properties. Within a few years of Conover's discovery, several semi-synthetic tetracyclines had entered the market, each with slightly different characteristics. This discovery established a paradigm that continues to dominate antibiotic development today. Most modern antibiotic discoveries are not entirely new molecular entities pulled fresh from nature, but rather novel active derivatives of older compounds, chemical variations on themes that nature had already composed. It was a fitting conclusion to this period of revolution. Humanity had learned not only to find antibiotics in nature, not only to produce them at industrial scale, but also to improve upon them through chemistry. By the mid-1950s, the transformation was complete. In less than 3 decades, medicine had progressed from having no effective antibacterial drugs to having a diverse arsenal including sulfonamides, penicillins, streptomycin, chloramphenicol, tetracyclines, and more. Diseases that had killed throughout human history could now be cured with a course of pills or injections. It seemed, for a brief shining moment, that humanity had conquered bacterial infections forever. What no one yet fully appreciated was that the bacteria were already beginning to fight back. To understand how antibiotics work their remarkable effects, we must first enter the microscopic world of the bacterial cell, a world of membranes and molecular machines, of carefully orchestrated chemical processes that sustain life at its most fundamental level. Each antibiotic we've discovered represents a different strategy for disrupting these processes, a different way of interfering with the bacterial machinery while leaving our own cells unharmed. Let's explore these elegant mechanisms, one by one. We begin with the most famous class of antibiotics, the beta-lactams, which include the penicillins that Fleming discovered, as well as cephalosporins, carbapenems, and monobactams. At the heart of each of these molecules lies a four-membered ring structure, the beta-lactam ring, that gives the class its name. This small chemical feature is the key to their antibacterial power. Bacteria, unlike our own cells, are surrounded by a rigid cell wall made of a substance called peptidoglycan. Think of it as a molecular mesh that encases the bacterium, giving it structural integrity and protecting it from bursting under osmotic pressure. The construction of this wall requires a precise sequence of enzymatic steps, and it's the final step, the cross-linking of peptide chains to form the mesh, that beta-lactams disrupt. The enzymes responsible for this final cross-linking are called penicillin-binding proteins, or PBPs, named for the first antibiotic found to bind to them. These enzymes, specifically transpeptidases and dealanil carboxypeptidases, catalyze the terminal transpeptidation process that joins peptides together to create the strong, interconnected peptidoglycan structure. The beta-lactam ring, with its strained four-membered structure, binds irreversibly to the active site of these enzymes, forming a stable complex that blocks their activity. But the damage doesn't end with simple inhibition. The antibiotic penicillin binding protein complex triggers something more dramatic. It stimulates the release of autolysins, enzymes that the bacterium normally uses to remodel its cell wall during growth. With wall synthesis blocked but autolysins still active, the existing cell wall begins to be digested from within. The bacterium loses its structural integrity and undergoes lysis. It essentially falls apart. This makes beta-lactam antibiotics bactericidal. They actively kill bacteria rather than merely inhibiting their growth. This mechanism is particularly elegant because it exploits a structure that human cells don't possess. We have cell membranes but no peptidoglycan cell walls, which is why beta-lactams can be given at high doses with relatively few side effects. They're attacking a target that simply doesn't exist in our own cells. Other antibiotics also target cell wall synthesis, but at different stages of the construction process. Vancomycin, a glycopeptide antibiotic that would become crucial in treating resistant infections, works earlier in the pathway. It binds to the dealanine termini, the ending portion of the peptidoglycan precursor molecules, preventing them from being incorporated into the growing cell wall. It's as if vancomycin intercepts the building blocks before they can be added to the structure, rather than inhibiting the enzymes that install them. Even earlier in the pathway, we find fosfomycin, an antibiotic with a unique mechanism. It targets an enzyme called UDP-N-acetylglucosamine enolpyravil transferase, known more simply as MURA. This enzyme catalyzes one of the very first steps in peptidoglycan synthesis, the condensation of phosphoenolpyruvate with UDP-N-acetylglucosamine to produce a molecule called UDP-MUR-NAC, an essential precursor for all peptidoglycan synthesis. Fosfomycin works by binding covalently, forming a permanent chemical bond, to a cysteine residue in the enzyme's active site, rendering it inactive. By blocking this early step, Phosphomycin prevents the bacterium from producing any of the building blocks needed for cell wall construction. Originally isolated from Streptomyces species, phosphomycin has proven particularly effective against urinary tract infections, and it has an additional benefit. It reduces bacterial adhesion to urinary epithelial cells, making it harder for bacteria to establish infections in the first place. Now let's turn our attention to a different cellular target: the bacterial ribosome, the molecular machine responsible for synthesizing proteins. Proteins are essential for virtually every cellular function, and disrupting their synthesis is a powerful way to stop bacterial growth. Several classes of antibiotics target the ribosome, but they do so in remarkably different ways. Aminoglycosides, a class that includes streptomycin, the first treatment for tuberculosis, as well as gentamicin, tobramycin, kanamycin, and neomycin, work by binding to the 30S ribosomal subunit, the smaller of the two parts that make up the bacterial ribosome. By binding there, they interfere with the accuracy of protein synthesis, causing the ribosome to misread the genetic code and incorporate incorrect amino acids into growing protein chains. The resulting misfolded, non-functional proteins accumulate in the bacterial cell, and the bacterium dies. This makes aminoglycosides bactericidal. They kill rather than merely inhibit. Interestingly, the nomenclature of aminoglycosides tells us something about their origin. Those derived from Streptomyces bacteria are given names ending in mycin, streptomycin, neomycin. Those derived from Micromonospora bacteria end in mycin, gentamicin, scysomycin. It's a small linguistic detail that reflects the diversity of nature's antibiotic-producing organisms. For gentamicin, researchers have identified specific pharmacodynamic parameters that predict success in treating bloodstream infections. When the ratio of the area under the concentration time curve to the minimum inhibitory concentration exceeds 70, or when the ratio of peak concentration to mic reaches 8 or higher, treatment outcomes improve significantly. This kind of precision in dosing reflects how far we've come in understanding not just what antibiotics do, but how to use them most effectively. Macrolide antibiotics, including erythromycin, azithromycin, and clarithromycin, also target protein synthesis, but they work on the larger 50S ribosomal subunit and employ a more subtle mechanism. For many years, scientists believed that macrolides simply blocked the nascent peptide exit tunnel, the channel through which newly synthesized proteins emerge from the ribosome. The image was straightforward. The antibiotic plugged the tunnel, and no proteins could be made. But recent structural and genomic studies have revealed something far more interesting. Macrolides don't completely block protein synthesis. They selectively inhibit the production of specific proteins. The growing protein chain can sometimes thread through the macrolide-obstructed tunnel, and whether it succeeds depends on the specific amino acid sequence being synthesized. In bacterial cells treated with erythromycin at concentrations 100 times higher than needed to inhibit growth, about 5 to 7% of total protein synthesis continues. Macrolides are, in essence, selective modulators rather than universal blockers. They prevent the synthesis of some proteins while allowing others to be made, and it's the loss of the critical proteins that ultimately stops bacterial growth. Linezolid represents an even more refined approach to inhibiting protein synthesis. Approved in 2000, it was the first member of the oxazolidinone class of antibiotics, and the first new class with a unique mechanism in decades. Rather than interfering with the elongation process like other protein synthesis inhibitors, linezolid prevents the very formation of the functional ribosome. It binds to the 23S ribosomal RNA of the 50S subunit, specifically at the peptidyl transferase center, and prevents the 30S and 50S subunits from coming together to form the functional 70S initiation complex. Without this complex, protein synthesis cannot even begin. This unique mechanism means linezolid doesn't compete with other antibiotics for binding sites, and it proved particularly valuable against methicillin-resistant Staphylococcus aureus and vancomycin-resistant Enterococci, bacteria that have become resistant to many other drugs. Its effectiveness extends to biofilm-forming strains, with minimum inhibitory concentrations typically ranging from just 0.5 to 4 micrograms per milliliter. Chloramphenicol, a broad-spectrum antibiotic originally isolated from the soil bacterium Streptomyces venezuelae and now produced synthetically, takes yet another approach to the ribosome. It binds reversibly to the L16 protein of the 50S subunit and suppresses the activity of peptidyl transferase, the enzyme that forms the peptide bonds linking amino acids together. By inhibiting this catalytic activity, chloramphenicol prevents the transfer of amino acids to growing peptide chains, effectively halting protein synthesis. Despite its effectiveness against a wide variety of microorganisms, chloramphenicol use has become limited by serious side effects. The drug can cause two types of bone marrow toxicity. The more common form is a dose-dependent, reversible suppression affecting up to half of patients who receive the drug, a consequence of mitochondrial injury that fortunately resolves when the drug is stopped. The second form is far more serious, a rare but potentially fatal aplastic anemia that occurs in approximately 1 in 25,000 to 40,000 patients. The mechanism remains incompletely understood but likely involves toxic metabolites produced when the drug's nitro group is chemically transformed in the body, leading to damage of bone marrow stem cells. Because of these risks, chloramphenicol is now usually reserved for life-threatening infections such as typhoid fever, where its benefits clearly outweigh the dangers. Moving beyond cell walls and ribosomes, we find antibiotics that target bacterial DNA itself. Quinolones and fluoroquinolones, including ciprofloxacin, levofloxacin, and moxifloxacin, represent broad-spectrum antibiotics that inhibit bacterial DNA synthesis by targeting two essential enzymes, DNA gyrase and topoisomerase 4. Both are type 2 topoisomerases, enzymes responsible for managing DNA topology during replication and transcription. As DNA unwinds during these processes, it becomes twisted and supercoiled, much like what happens when you unwind a twisted rope. DNA gyrase relieves this tension by temporarily cutting both strands of the DNA double helix, passing another segment of DNA through the break, and then resealing it, a remarkable molecular gymnastics that allows replication to proceed. Topoisomerase 4 performs similar functions, particularly in separating interlinked daughter chromosomes after DNA replication. Fluoroquinolones don't simply inhibit these enzymes, they convert them into cellular poisons. The drugs stabilize the cleavage complex, the moment when the enzyme has cut the DNA but hasn't yet resealed it. Trapped in this state with broken DNA, the bacterium cannot complete replication and ultimately dies. Research has shown that ciprofloxacin interacts with both the enzyme and DNA through a water-metal ion bridge, a structural feature that anchors the drug in place. Mutations in the amino acid residues that form this bridge, particularly in regions called quinolone resistance determining regions, diminish the drug's effectiveness by weakening its ability to stabilize the cleavage complex. For anaerobic bacteria and certain protozoa, metronidazole offers a completely different mechanism. This nitroimidazole antibiotic enters cells through passive diffusion and undergoes reduction of its nitro group, but only in organisms with very low oxygen levels and specific electron transport proteins like ferredoxin. This reduction produces reactive intermediates, nitro-radical anions, that directly inhibit DNA synthesis. What's particularly interesting is that metronidazole doesn't break DNA strands or directly inhibit DNA polymerase. Instead, it causes an immediate cessation of DNA synthesis by blocking the replication machinery from copying the genetic code. The DNA remains structurally intact, but the bacterium can no longer replicate it. This selective activation in anaerobic environments means metronidazole is highly effective against obligate anaerobes, organisms that can only survive without oxygen, including Bacteroides fragilis, Clostridium species, and protozoa like Giardia and Entamoeba, while having minimal effect on oxygen-using bacteria or human cells. Some antibiotics employ mechanisms so specialized that they target only specific pathogens. Fidaxomicin, approved in 2011, exemplifies this approach. It's designed specifically for Clostridioides difficile infections, one of the most common healthcare-associated infections. Unlike broad-spectrum antibiotics that devastate beneficial gut bacteria along with pathogens, fidaxomicin has a narrow spectrum. It selectively targets C. difficile while largely sparing the innocent bacterial bystanders that make up our healthy microbiome. Fidaxomicin inhibits bacterial RNA polymerase, the enzyme that transcribes DNA into RNA, but it does so in a unique way. The drug binds at the base of the enzyme's clamp, a movable part of the polymerase that must close around DNA for transcription to occur. Fidaxomicin wedges into the hinge between two subunits, jamming the clamp open and preventing the enzyme from grabbing onto genetic material. Critically, it can only block initiation if added before the polymerase forms the open promoter complex, the configuration where DNA strands have separated and transcription is ready to begin. Once this complex forms, fidaxomicin cannot intervene. This specificity for the initiation step, combined with structural features that make it selective for C. difficile RNA polymerase over those of other bacteria, gives fidaxomicin its narrow spectrum. As an additional benefit, it inhibits spore production in C. difficile, which may explain why patients treated with fidaxomicin experience fewer recurrences than those treated with vancomycin, the previous standard therapy. Finally, we come to daptomycin, a calcium-dependent cyclic lipopeptide antibiotic approved in 2003, which has revealed layers of complexity that scientists are still working to fully understand. For years, the mechanism seemed straightforward. Daptomycin disrupts bacterial cell membranes, causing them to depolarize and lose their electrical potential, leading to cell death. But recent research in 2025 has revealed that daptomycin actually operates through two independent mechanisms. The first mechanism, membrane depolarization, occurs when daptomycin interacts with regular membrane lipids. This effect doesn't require the bacteria to be actively growing. It works even on resting cells. The second mechanism involves the formation of lipid domains, but this only occurs when daptomycin interacts with specific cell wall precursor lipids, particularly molecules like lipid 2 that are involved in peptidoglycan synthesis. This second mechanism is limited to actively growing cells. Earlier research had shown that daptomycin causes profound reorganization of membrane architecture, leading to mislocalization of essential cell division proteins like DIVIVA. When these proteins end up in the wrong place, cell wall synthesis and septation, the process of dividing into two daughter cells, are severely disrupted. The drug requires calcium ions to function, using them to bind to phosphatidylglycerol in bacterial membranes. Once bound, daptomycin molecules oligomerize, coming together to form larger structures that cause the membrane damage. This dual mechanism makes daptomycin particularly effective against gram-positive pathogens, including methicillin-resistant Staphylococcus aureus, and helps explain its continued effectiveness even as bacteria develop resistance to other drugs. These mechanisms, targeting cell walls, ribosomes, DNA, RNA, and membranes, represent the major strategies that antibiotics employ. Each one exploits a difference between bacterial cells and our own, finding vulnerabilities in bacterial biology that our cells don't share. It's a testament to both the diversity of life and the ingenuity of the researchers who unraveled these molecular details. But as we'll see in the sections ahead, bacteria are not passive victims. They have evolved, and continue to evolve, equally elegant ways to resist these chemical attacks. Before we explore how bacteria develop resistance to antibiotics, we must first understand that not all bacteria are equally vulnerable to begin with. Long before the advent of antibiotics, bacteria had evolved structural features that would later prove to be natural defenses. To appreciate these differences, we turn to a staining technique developed in the 19th century that would become one of the most important tools in microbiology. In 1884, a Danish bacteriologist named Hans Christian Gram developed what he initially thought was simply a better way to visualize bacteria under the microscope. He treated bacterial samples with crystal violet dye, followed by iodine, then attempted to wash away the stain with alcohol, and finally applied a contrasting red dye called safranine. What he discovered was that bacteria didn't all respond the same way. Some retained the purple crystal violet stain even after the alcohol wash, while others lost the purple and took up the pink safranine counterstain instead. Gram had inadvertently created not just a staining technique, but a fundamental classification system that revealed deep structural differences between bacterial groups. We now call these gram-positive and gram-negative bacteria, and the distinction runs far deeper than mere color under a microscope. It reflects profound differences in cell wall architecture that have enormous implications for antibiotic susceptibility. Gram-positive bacteria retain the crystal violet stain because they possess a thick layer of peptidoglycan, that molecular mesh we discussed earlier, that makes up approximately 90% of their cell wall. This peptidoglycan layer is heavily cross-linked, creating a dense but porous structure that surrounds the bacterial cell membrane. When the crystal violet iodine complex forms during gram staining, it becomes trapped in this thick peptidoglycan matrix and cannot be washed away by alcohol. Gram-negative bacteria, on the other hand, have a much thinner peptidoglycan layer with less cross-linkage. But what they lack in peptidoglycan thickness, they compensate for with an additional outer membrane, a second lipid bilayer that lies outside the thin peptidoglycan layer. This outer membrane contains lipopolysaccharides and proteins, creating an extra barrier that gram-positive bacteria don't possess. During gram staining, the thin peptidoglycan layer cannot retain the crystal violet iodine complex once alcohol disrupts the outer membrane, so these bacteria lose the purple stain and take up the pink safranine counterstain instead. These structural differences have profound implications for how antibiotics work. The thick, porous peptidoglycan layer of gram-positive bacteria allows antibiotics relatively easy access to their targets. Drugs can diffuse through this mesh-like structure, reaching the cell membrane and interior without significant obstruction. This is why many antibiotics that target peptidoglycan synthesis or that need to reach the cell membrane are particularly effective against gram-positive bacteria. Gram-negative bacteria present a far greater challenge. Their outer membrane acts as an effective barrier, regulating the passage of large molecules, including many antibiotics, into the cell. This membrane is not just a passive barrier but a selective one, studded with protein channels called porins that control what enters. The lipopolysaccharide component creates a hydrophobic region that many drugs struggle to cross, while the thin peptidoglycan layer, though present, offers less of a target for drugs that disrupt cell wall synthesis. This structural difference explains patterns we see clinically. Penicillin, for instance, works wonderfully against many gram-positive bacteria but is far less effective against gram-negatives. Not because the target, penicillin-binding proteins, is absent, but because the drug has difficulty reaching that target through the outer membrane. This is why, as antibiotic development progressed, researchers specifically designed drugs with features that would help them penetrate gram-negative bacteria, leading to the development of so-called broad-spectrum antibiotics effective against both gram-positive and gram-negative organisms. But even beyond these structural differences, all bacteria possess what we call intrinsic antibiotic resistance, a naturally occurring phenomenon that predates the medical use of antibiotics by millions of years. This intrinsic resistance, as scientists call it, is present in all bacterial species and represents the baseline level of resistance that exists simply as a consequence of bacterial biology. Three primary mechanisms contribute to this intrinsic resistance. First, bacteria may simply lack the target that a particular antibiotic is designed to attack. An antibiotic that inhibits a specific enzyme cannot harm a bacterium that doesn't possess that enzyme. Second, bacteria may produce chromosomally encoded enzymes that inactivate certain antibiotics, not because they evolved specifically to resist our drugs, but because these enzymes serve other cellular functions that happen to degrade or modify antibiotic molecules. Third, and perhaps most importantly, bacteria may have reduced uptake of antibiotics due to the permeability characteristics of their cellular envelopes or the activity of efflux pumps, molecular machinery that actively transports substances out of the cell. This last mechanism deserves particular attention, especially when we consider gram-negative bacteria. Pseudomonas aeruginosa, a notorious pathogen responsible for many hospital-acquired infections, exemplifies how intrinsic resistance can make treatment challenging. This bacterium has an outer membrane with permeability up to 100 times lower than that of Escherichia coli. Combined with numerous efflux pumps that actively expel antibiotics, this creates a formidable natural defense that contributes significantly to Pseudomonas's reputation for being difficult to treat. What's crucial to understand is that the intrinsic resistance, this collection of resistance elements that bacteria naturally possess, did not evolve specifically to counteract modern antibiotics. Rather, many of these elements are involved in basic bacterial metabolic processes, performing essential cellular functions that incidentally provide resistance. The characteristic antibiotic susceptibility pattern of any given bacterial species emerges from the combined activity of these various elements, each contributing its share to the overall resistance profile. The challenge of antibiotic penetration through gram-negative outer membranes has become one of the central problems in antibiotic development. Most successful antibiotics that work against gram-negative bacteria enter through nonspecific porins, protein channels like OMP-F and OMP-C in E. coli that normally allow hydrophilic metabolites to pass through the membrane. These porins aren't designed to let antibiotics through. Rather, antibiotics must exploit these existing channels if they're to reach their intracellular targets. Recent computational and experimental studies have revealed the molecular details of how some antibiotics manage this feat. Many successful antibiotics contain a positively charged amine group, and research has shown that this positive charge interacts favorably with negative charges lining the interior of bacterial porins. As the antibiotic molecule threads through the narrowest part of the channel, the constriction zone, this favorable interaction allows it to line up in an energetically favorable manner, facilitating passage through what would otherwise be an impenetrable barrier. Scientists have confirmed this mechanism through elegant mutation experiments. When they altered the negatively charged amino acids lining the porin, the interaction with the antibiotic's amine group was disrupted, and the drug could no longer pass through efficiently. This understanding has opened new possibilities. By designing antibiotics with appropriate chemical features, or by modifying existing drugs to enhance their interaction with porins, researchers can potentially improve penetration into gram-negative bacteria and overcome one of nature's most effective antibiotic barriers. This interplay between bacterial structure and antibiotic access, between the natural features bacteria evolved over billions of years and the drugs we developed over mere decades, sets the stage for understanding acquired resistance. The intrinsic defenses we've explored here are just the beginning. As we'll see, bacteria can build upon these natural foundations, acquiring new mechanisms that transform manageable infections into deadly threats. But first, we must understand how these acquired resistance mechanisms work at the molecular level. Now we arrive at one of the most sobering chapters in our story: the rise of acquired antibiotic resistance. If intrinsic resistance represents bacteria's natural baseline defenses, acquired resistance is their evolutionary response to the antibiotics we've deployed against them. What we're witnessing is adaptation in real time, a demonstration of evolution's power that unfolds not over millions of years but across mere decades, sometimes even within the course of a single patient's treatment. Bacteria employ 4 primary strategies to overcome antibiotics, and together these mechanisms explain most of the resistance we encounter clinically. Understanding them is essential not just for appreciating the scope of the problem, but for grasping why certain treatment approaches succeed while others fail. The first strategy is enzymatic inactivation. Bacteria producing enzymes that chemically modify or destroy antibiotics before they can reach their targets. The most famous examples are the beta-lactamases, enzymes that break down beta-lactam antibiotics by cleaving that crucial 4-membered ring we discussed earlier. Once the ring is opened, the antibiotic loses its ability to bind to penicillin-binding proteins, rendering it harmless to the bacterium. Beta-lactamases have diversified into hundreds of variants, each with slightly different substrate preferences. KPC, NDM, and OXA48 variants represent particularly concerning carbapenemases, beta-lactamases that can destroy even carbapenems, the antibiotics we often reserve as last resort treatments for resistant infections. The second strategy involves efflux pumps, molecular machines embedded in bacterial membranes that actively expel antibiotics from the cell. These aren't new inventions created in response to antibiotics. Many efflux pumps serve normal cellular functions, removing toxic metabolites and other unwanted substances. But bacteria can overexpress these pumps, producing many more copies than normal, turning what was a housekeeping function into an antibiotic resistance mechanism. Pumps like ADA-ABC in Acinetobacter, ACRA-B-Tol-C in E. coli, and Mexabioprim in Pseudomonas aeruginosa can transport multiple classes of antibiotics out of the cell, making these bacteria resistant to several drugs simultaneously, a phenomenon called multi-drug resistance. The third strategy is target site modification. Bacteria altering the very molecules that antibiotics are designed to attack. If an antibiotic binds to a specific site on a ribosomal protein, a mutation that changes even a single amino acid at that site can prevent binding while still allowing the ribosome to function. Modifications to penicillin-binding proteins, alterations in RNA polymerase, changes to ribosomal proteins, mutations in the quinolone resistance determining regions of DNA gyrase. All of these represent molecular tweaks that maintain bacterial function while undermining antibiotic effectiveness. The fourth strategy is reduced membrane permeability, making it harder for antibiotics to enter the bacterial cell in the first place. Bacteria can downregulate or modify their porins, those protein channels through which many antibiotics must pass. They can thicken their outer membranes or change their composition to make them less permeable. By reducing the amount of antibiotic that reaches the inside of the cell, bacteria can survive concentrations that would normally kill them. What makes resistance particularly concerning is that these mechanisms often combine. A bacterium might overexpress efflux pumps while simultaneously producing beta-lactamases and modifying its porins. This layering of defenses explains why some bacteria become resistant to nearly every available antibiotic. They've accumulated multiple independent mechanisms, each providing a degree of protection, and together creating nearly impenetrable armor. Evidence from research conducted as recently as 2026 demonstrates that resistance emerges rapidly even against newly approved antibiotics. What's particularly striking is that bacteria often repurpose pre-existing genetic pathways, adapting ancient resistance mechanisms to work against new chemical structures. The speed of this adaptation suggests that bacteria have a vast reservoir of potential resistance mechanisms waiting to be activated or modified when they encounter new threats. Among these mechanisms, efflux pumps deserve special attention because of their role in multi-drug resistance. The resistance nodulation division family of efflux pumps, found in gram-negative bacteria, demonstrates particularly broad substrate specificities, meaning a single pump can expel many structurally different drugs. In Pseudomonas aeruginosa, the MEX-A-B-Operam efflux pump can transport beta-lactams, fluoroquinolones, macrolides, and sulfonamides, among others. A bacterium expressing this pump at high levels gains resistance to multiple antibiotic classes simultaneously. Recent studies suggest that efflux pumps play critical but underappreciated roles in resistance to newer beta-lactams and beta-lactam/beta-lactamase inhibitor combinations, antibiotics specifically designed to overcome earlier resistance mechanisms. Mutations in pump components or in the regulatory genes that control pump expression have been associated with increased minimum inhibitory concentrations for these novel antibiotics. When resistant bacterial lineages are examined, increased expression of pump components is consistently found, demonstrating that even as we develop new drugs, bacteria can adapt their efflux mechanisms to defend against them. The story of vancomycin resistance illustrates how bacteria can evolve remarkably sophisticated countermeasures. Vancomycin, introduced clinically in the late 1950s, works by binding to the dealanine-dealanine terminus of peptidoglycan precursors, preventing cell wall synthesis. For 3 decades, high-level resistance to vancomycin seemed impossible. The drug interacted with its target through 5 hydrogen bonds, creating a very strong and specific interaction. Then, in 1988, high-level vancomycin resistance appeared in enterococci. The VANA resistance mechanism that emerged involved replacing the terminal dealanine-dealanine with dealanine-D-lactate, swapping one amino acid for a similar but subtly different molecule. This simple substitution had profound effects. It removed one of the 5 hydrogen bonds that vancomycin forms with its target and introduced a repulsive interaction between oxygen atoms that actually pushes the drug away. The result was a 1,000-fold loss in vancomycin's binding affinity. The drug could no longer stick to its target effectively, and the bacteria became resistant. By 2002, vancomycin resistance had jumped to Staphylococcus aureus, creating VRSA, vancomycin-resistant Staph aureus. The mechanism was the same VANA gene cluster, but the route of transmission told an important story about how resistance spreads. S. aureus had acquired the VANA genes from vancomycin-resistant enterococci through plasmid-mediated horizontal gene transfer, one species sharing its resistance genes with another through mobile genetic elements we'll explore more deeply in the next section. Perhaps most remarkably, Researchers have discovered glycopeptide resistance genes in ancient DNA recovered from 30,000-year-old permafrost. This finding demonstrates that glycopeptide resistance, and antibiotic resistance more generally, actually predates modern clinical antibiotic use. These resistance genes existed in environmental bacteria long before we began using antibiotics as medicines, presumably conferring protection against naturally occurring antimicrobial compounds produced by other microorganisms. What modern antibiotic use has done is create selective pressure that favors bacteria carrying these genes, causing resistance to spread and become more common. Quinolone resistance provides another clear example of how target modification confers resistance. Fluoroquinolones like ciprofloxacin kill bacteria by stabilizing the DNA gyrase DNA cleavage complex, as we discussed earlier. The drugs interact with both the enzyme and DNA through a water-metal ion bridge, and this interaction depends on specific amino acid residues in the quinolone resistance determining regions of the genes encoding DNA gyrase and topoisomerase 4. Mutations in these regions, particularly in the GYRA and GYRB genes for DNA gyrase and the PARC and PAIR genes for topoisomerase 4, can dramatically reduce drug susceptibility. A single amino acid substitution, such as changing serine to leucine at position 83 or aspartic acid to asparagine at position 87 in E. coli GYRA, can increase the minimum inhibitory concentration 8 to 32-fold. When mutations accumulate in both GYRA and PARC, resistance reaches high levels. These mutations work by disrupting the amino acid residues that anchor the water-metal ion bridge, weakening the drug's ability to stabilize the cleavage complex. The gyrase can still perform its normal function of managing DNA topology, but the drug can no longer convert it into a cellular poison. Tetracycline resistance reveals yet another elegant bacterial countermeasure through ribosomal protection proteins. Unlike typical resistance mechanisms, these proteins don't prevent tetracycline from entering the cell or destroy it chemically. Instead, they bind directly to bacterial ribosomes and actively displace tetracycline from its binding site on the 30S ribosomal subunit. Ribosomal protection proteins like TET-O and TET-M are translational GT-PASAs, enzymes that use GTP hydrolysis for energy, with significant structural similarity to the elongation factor EFG, which normally helps move the ribosome along messenger RNA during protein synthesis. These protection proteins essentially mimic a normal cellular component, allowing them to interact productively with the ribosome. When tetracycline binds to the ribosome and blocks protein synthesis, these protection proteins bind in a GTP-dependent manner and use conformational changes to pry the antibiotic off. Structural studies have revealed that TET-O positions 3 characteristic loops in its domain 4 structure to directly compete with tetracycline at the binding site. Mutations in these critical loops result in complete or near-complete loss of protective activity, confirming their importance. The genes encoding these proteins have evolved into at least 12 distinct classes and have spread through horizontal genetic transfer into a wide range of bacteria, with TET-M becoming the predominant ribosomal protection protein gene found in Escherichia coli. The development of beta-lactamase inhibitors represents one of medicine's attempts to fight back against enzymatic inactivation. Clavulanic acid, discovered in the 1970s, was the first of these inhibitors and revolutionized the treatment of beta-lactamase-producing bacteria. Produced naturally by Streptomyces clavuligerus, clavulanic acid is itself a beta-lactam compound, containing the characteristic 4-membered ring. But rather than acting as an antibiotic, it acts as a suicide inhibitor of beta-lactamase enzymes. The mechanism is elegant in its molecular detail. Clavulanic acid's carboxyl group at position 7 forms a hydrogen bond with a serine residue, specifically serine 70, in the active site of beta-lactamase. This interaction favors the formation of a stable acyl intermediate through nucleophilic attack, just as would happen with a normal beta-lactam antibiotic. But then something different occurs. The 5-membered oxazolidine ring opens, and the inhibitor linearizes, generating a cis-amine structure. This structure can isomerize to an even more stable transamine. Through this series of chemical rearrangements, clavulanic acid becomes irreversibly bound to the beta-lactamase, permanently inactivating it. When combined with beta-lactam antibiotics like amoxicillin, in formulations like Augmentin, clavulanic acid protects the antibiotic from destruction. The beta-lactamase enzymes bind to and are inactivated by the clavulanic acid, allowing the amoxicillin to reach its targets and kill the bacteria. This combination restored effectiveness against many beta-lactamase-producing organisms, including certain strains of Staphylococcus aureus, E. coli, and Haemophilus influenza. Yet even this clever solution has its limitations. Not all beta-lactamases are inhibited by clavulanic acid, particularly the metallo-beta-lactamases like NDM that we mentioned earlier, which use a zinc ion in their active site rather than a serine residue. And over time, bacteria have evolved new beta-lactamases that are less susceptible to inhibition. The arms race continues, with each innovation on our part eventually met by bacterial counter-innovation. As we reflect on these mechanisms, enzymes that destroy drugs, pumps that expel them, targets that change shape, and proteins that remove them from their binding sites, a pattern emerges. Bacteria are not passive victims of our interventions, but active participants in an evolutionary contest. Every antibiotic we deploy represents a new selective pressure, and bacteria, with their rapid reproduction and vast populations, can explore genetic space far faster than we can develop new drugs. Understanding these mechanisms in detail is the first step toward developing strategies that might slow or circumvent resistance. But as we'll see in the next section, the problem is compounded by bacteria's ability to share these resistance mechanisms with one another. If bacteria could only pass resistance genes to their offspring, vertically, from parent to daughter cell, the problem of antibiotic resistance would be far more manageable. Each resistant strain would remain isolated, confined to its own lineage. But bacteria possess something that makes them far more formidable: the ability to share genetic information horizontally, transferring resistance genes not just to their descendants but to their neighbors, even to entirely different species. This horizontal gene transfer is the main cause of bacterial antibiotic resistance, and it enables a form of collaboration in resistance development that greatly accelerates the spread of defensive mechanisms across the microbial world. Imagine a bacterial population as a community where useful information can be shared rapidly between members, regardless of their relationship. A bacterium that has developed resistance to an antibiotic through mutation can transfer that capability to other bacteria around it, which can then transfer it onward, creating a cascade of resistance that spreads through the population far faster than vertical inheritance alone would allow. This genetic sharing fundamentally changes the dynamics of resistance evolution and explains why resistance can emerge and spread with such alarming speed. 3 primary mechanisms enable this horizontal transfer, each with its own molecular machinery and characteristics. The first and most important for spreading antibiotic resistance is conjugation, direct transfer of DNA through cell-to-cell contact. The second is transformation, in which bacteria take up naked DNA from their environment, DNA that might have been released from dead cells or secreted by living ones. The third is transduction, where bacteriophages, viruses that infect bacteria, accidentally package bacterial DNA and transfer it when they infect new hosts. Scientists have also verified that membrane vesicles, small bubble-like structures that bacteria release, can play roles in horizontal gene transfer, adding yet another route for genetic exchange. Each of these mechanisms contributes to resistance spread, but conjugation through plasmids plays a particularly major role in the dissemination of antimicrobial resistance. To understand why, we need to understand what plasmids are and how they move between cells. Plasmids are small, circular pieces of DNA that exist separately from the main bacterial chromosome. They can replicate independently, making copies of themselves inside the bacterial cell. While the chromosome contains the essential genes needed for basic bacterial survival and reproduction, plasmids often carry accessory genes, genetic information that isn't strictly necessary but can provide advantages under certain conditions. Antibiotic resistance genes are frequently found on plasmids, making these mobile genetic elements prime vehicles for spreading resistance. Most resistance plasmids are conjugative, meaning they encode all the components needed for their own transfer to another bacterium. The process begins when a donor cell extends a structure called a pilus, a tube-like appendage that reaches out and makes contact with a recipient cell. Once contact is established, the plasmid DNA is copied, and one copy is transferred through this bridge into the recipient. After transfer, both cells possess the plasmid, and both can now act as donors, potentially transferring the plasmid to other cells they encounter. What makes this particularly concerning is that plasmids often carry multiple resistance genes simultaneously. A single plasmid might encode resistance to 3, 4, 5, or more different antibiotics. When such a plasmid transfers to a new host, that host immediately gains resistance to all those antibiotics at once. Instant multi-drug resistance without having to evolve each resistance mechanism independently. This explains why we sometimes see bacteria that are suddenly resistant to multiple drugs they've never encountered before. They've received a resistance plasmid from another bacterium that had already accumulated those genes. Research has revealed something even more surprising about plasmid transfer. It can occur even without antibiotic selective pressure. Traditional thinking held that bacteria would only maintain and transfer resistance plasmids when antibiotics were present, because carrying these extra genes should impose a metabolic cost. Why maintain machinery you don't need? But studies have demonstrated that plasmids can be transferred between bacteria even in antibiotic-free environments, meaning there may be no directly visible fitness advantage in acquiring the resistance-carrying plasmid. The transfer happens anyway, possibly maintained by other selective pressures we don't fully understand, or simply because the cost of carrying the plasmid is low enough that it persists even without conferring immediate benefits. Even more paradoxically, antibiotics themselves can increase the rate of plasmid transfer. Researchers have found that subinhibitory concentrations of antibiotics, levels below what's needed to kill bacteria or completely stop their growth, can upregulate the transfer genes in bacteria. Studies have shown that antibiotics including cefotaxime, ampicillin, and ciprofloxacin increase the expression of TRA genes, the genes encoding the transfer machinery for conjugation. This means that even when we use antibiotics at concentrations too low to kill bacteria, perhaps in the environment where antibiotics from farms or hospitals have been diluted, or in body sites where drug concentrations are suboptimal, we're actually promoting the spread of resistance genes. The antibiotics accelerate their own obsolescence. Beyond plasmids, another class of mobile genetic elements plays a crucial role in resistance spread: transposons. These are DNA sequences that can move within and between DNA molecules, earning them the nickname jumping genes. Transposons don't replicate independently like plasmids. Instead, They insert themselves into other pieces of DNA, chromosomes, plasmids, or other mobile elements. Transposons carrying antibiotic resistance genes can move from a chromosome to a plasmid, or from one plasmid to another, or from a plasmid into a chromosome. This mobility accelerates antibiotic resistance dissemination in microbial communities because it means resistance genes aren't permanently tied to one genetic context. A resistance gene that starts on the bacterial chromosome can jump to a conjugative plasmid and suddenly gain the ability to spread to other cells. A gene on one plasmid can jump to another plasmid that has a broader host range, allowing it to move into different bacterial species. Mathematical modeling and experimental evolution demonstrate that antibiotic selection drives the evolution of duplicated resistance genes through transposon-mediated intrachromosomal transposition events. Transposons copying resistance genes and inserting additional copies elsewhere in the same chromosome. In laboratory evolution experiments, researchers observed antibiotic resistance gene duplications in 100% of evolved bacterial populations across all 4 tested antibiotic treatments. More copies of a resistance gene mean more of the resistance protein being produced, which can increase the level of resistance. When researchers analyzed 18,938 complete bacterial genomes, they found that duplicated antibiotic resistance genes are highly enriched in bacteria isolated from humans and livestock compared to environmental isolates. This indicates ongoing positive selection in environments associated with antibiotic use. Wherever antibiotics are commonly deployed, bacteria with duplicated resistance genes have an advantage and become more common. The transposons that carry these genes have become vehicles for amplifying resistance. Examples like TN1721, a transposon that carries genes conferring resistance to beta-lactam antibiotics, demonstrate how these elements facilitate rapid spread across bacterial populations. Once a resistance gene becomes associated with a transposon, it gains mobility that allows it to spread far beyond its original host. Recent research has revealed something even more complex: synergistic horizontal transfer of antibiotic resistance genes and specific transposons. A study published in 2024 focused on infant gut microbiota and identified that certain bacterial species play central roles in the synergistic transfer, particularly Klebsiella pneumoniae, Enterobacter hormicii, and Escherichia coli. These species act as hubs in the resistance gene network, both acquiring and distributing resistance determinants. What's particularly striking is that infant gut microorganisms appear more susceptible to horizontal gene transfer events involving specific antibiotic resistance genes and transposons compared to adult gut microorganisms. This suggests age-related differences in susceptibility to resistance gene acquisition, possibly because the infant gut microbiome is less stable and more open to colonization by new genetic elements. The study revealed specific pairings, certain resistance genes traveling together with certain transposons more often than would be expected by chance, suggesting that these combinations are particularly successful at spreading. This brings us to one of the most sophisticated systems bacteria have evolved for acquiring and expressing multiple resistance genes: integrons. These genetic elements contain a site-specific recombination system that enables bacteria to capture, express, and exchange specific DNA fragments called gene cassettes. Think of an integron as a genetic platform with standardized connection points that allow plug-and-play addition of new genes. The structure of an integron includes an integrase gene called INTI, which encodes the enzyme that catalyzes the recombination reactions, a recombination site called ATTI, where new cassettes can be inserted, and a promoter called PC, which drives expression of the captured gene cassettes. Gene cassettes themselves are small mobile elements consisting of little more than a single gene and a recombination site called ATTC. Importantly, These cassettes typically lack their own promoters. They rely on the integron's promoter for expression, which means they only become functional once integrated into an integron. The elegance of this system lies in its modularity. Multiple gene cassettes can be inserted into the same integron, forming tandem arrays, a string of resistance genes lined up one after another, all under control of the same promoter. This allows bacteria to accumulate resistance to multiple antibiotics in a single genetic locus. The integrase can excise cassettes as well as insert them, meaning the system is dynamic. Cassettes can be reshuffled, lost, or added depending on selective pressures. More than 130 distinct antibiotic resistance genes have been identified within gene cassettes, conferring resistance to an impressive array of drugs: beta-lactams, aminoglycosides, chloramphenicol, streptothricin, trimethoprim, rifampin, erythromycin, quinolones, fosfomycin, lincomycin, and even quaternary ammonium compounds used as antiseptics and disinfectants. Most Class I integrons, the most clinically relevant type, also include a sulfonamide resistance determinant in their backbone structure, providing yet another layer of resistance. Gene cassettes can be integrated not only into integrons but also at secondary sites in bacterial genomes or on broad host range plasmids, further increasing their mobility. Single cassettes can move independently, or multiple cassettes can move as a unit. Although integrons themselves lack mobility functions, they cannot move between cells on their own. They are typically found on plasmids or transposons, which provide the mobility. This combination of integrons on mobile genetic elements creates one of the most significant horizontal transfer pathways for resistance genes. An updated compilation of gene cassettes found in mobile resistance integrons reveals the extraordinary diversity of this system. At least 130 different cassettes carrying known or predicted antibiotic resistance genes have been identified, along with many cassettes of unknown function. This library of resistance determinants represents a reservoir that bacteria can draw upon, mixing and matching resistance genes to create combinations suited to whatever antibiotic challenges they face. The implications are sobering. Through conjugation, transformation, and transduction, mediated by plasmids, transposons, and integrons, bacteria have created a genetic commons, a shared pool of resistance genes that can be accessed by diverse species. A resistance gene that evolves in one bacterial species in one part of the world can, through these mechanisms, end up in a completely different species on another continent. The boundaries between bacterial strains and species become porous when it comes to resistance genes. This horizontal transfer means that our efforts to contain resistance must consider not just preventing the evolution of new resistance mechanisms, but also preventing the spread of existing ones. An antibiotic resistance gene is not just a problem for the bacterial population where it arose, it's a potential problem for bacterial populations everywhere, because mobility is built into the system. As we move forward in our story, this understanding of how resistance spreads will inform our appreciation of why the problem has become so difficult to control and why comprehensive, coordinated approaches are necessary to address it. We've explored how bacteria develop resistance through genetic changes and share those changes through mobile genetic elements. But there's another dimension to bacterial survival that operates at a different scale. Not at the level of individual cells or even populations, but at the level of organized communities. Here, bacteria reveal themselves to be far more sophisticated than we once imagined, capable of communication, coordination, and collective behaviors that dramatically increase their ability to withstand antibiotic assault. When we think of bacteria, we often picture them as individual cells floating freely through liquids, what microbiologists call planktonic cells. And indeed, many bacteria do exist in this state, drifting through bloodstreams, settling onto surfaces, moving through water and soil. But this free-floating lifestyle is only part of the bacterial story. Given the opportunity, many bacteria prefer to settle down, attach to surfaces, and form structured communities encased in a self-produced matrix. These surface-attached communities are called biofilms, and they represent one of the most formidable challenges in treating bacterial infections. Picture the film that forms on river rocks, making them slippery underfoot. Imagine the plaque that accumulates on teeth despite brushing. Think of the coating that develops inside water pipes or on the hulls of ships. These are all biofilms, communities of microbial cells embedded in an extracellular matrix they've produced, creating structures that can be remarkably resistant to chemical and physical disruption. Within a biofilm, bacteria are significantly less susceptible to antimicrobial agents than their planktonic counterparts, sometimes requiring antibiotic concentrations hundreds of times higher to achieve the same killing effect. Several mechanisms contribute to this biofilm-based resistance, and together they create a protective environment that allows bacteria to survive treatments that would easily kill them in their free-floating form. The first mechanism involves the biofilm matrix itself, that extracellular material the bacteria produce and surround themselves with. This matrix, composed of exopolysaccharides, proteins, and extracellular DNA, acts as a physical barrier that antimicrobials must penetrate before reaching the bacterial cells. Some antibiotics bind to components of the matrix and never reach the bacteria at all. Others are slowed or inactivated as they diffuse through it. Extracellular DNA incorporated into Pseudomonas aeruginosa biofilms, for instance, can increase resistance threefold for the aminoglycoside tobramycin and twofold for gentamicin. The DNA molecules, carrying negative charges, can bind to positively charged antibiotics and sequester them, preventing them from reaching their targets. Other matrix components can have similar effects, creating a gauntlet that antibiotics must run before encountering any bacterial cells. But the matrix is just the beginning. Within biofilms, bacteria grow more slowly than their planktonic relatives, and this reduced growth rate itself provides protection. Many antibiotics, particularly beta-lactams and aminoglycosides, work best against actively growing cells. They target processes like cell wall synthesis or protein production, processes that are most active when bacteria are dividing rapidly. In the depths of a biofilm, where nutrients may be limited and waste products accumulate, bacterial growth slows. In this metabolically quiescent state, bacteria become less susceptible to antibiotics that rely on attacking growth and division. There's also a more insidious mechanism at work. Bacteria in biofilms can secrete enzymes that degrade antibiotics, beta-lactamases and other resistance enzymes we've discussed, and these enzymes remain active within the biofilm matrix. In biofilms formed by pathogens like Klebsiella pneumoniae and P. aeruginosa, secreted beta-lactamases effectively degrade antibiotics before they reach the cells that produce them, and even protect neighboring cells that might not produce the enzymes themselves. It's a form of cooperative defense, where the enzymatic activity of some cells shields the entire community. Beyond these mechanisms, biofilms harbor specific genetic determinants of antibiotic resistance and tolerance, genes that are specifically upregulated in the biofilm lifestyle, contributing additional layers of protection. The combination of physical barriers, slow growth, enzymatic degradation, and genetic adaptations makes biofilms extraordinarily difficult to eradicate with antibiotics alone. But how do bacteria coordinate the formation of these complex structures? How do individual cells know when to transition from free-floating existence to community living? The answer lies in a remarkable system of bacterial communication called quorum sensing, a process that allows bacteria to sense and respond to population density. Quorum sensing is, at its heart, a bacterial census system. As bacterial populations grow, individual cells release small signaling molecules called autoinducers. These molecules diffuse through the environment, and all nearby bacteria of the same species can detect them. When the concentration of autoinducers reaches a critical threshold, indicating that the population has reached a certain density, or quorum, bacteria respond by changing their gene expression patterns, coordinating colony-wide behaviors. The molecular details vary between bacterial groups. Gram-negative bacteria typically use acylhomoserine lactones as autoinducers, which are detected by LuxR-type receptors inside cells. Gram-positive bacteria often rely on oligopeptides, short chains of amino acids, that activate two-component signal transduction systems on the cell surface. Despite these differences in molecular machinery, the principle remains the same. Bacteria count their neighbors using chemical signals and respond collectively when they reach sufficient numbers. Quorum sensing coordinates a remarkable array of behaviors. It regulates sporulation, the formation of hardy, dormant spores that can survive extreme conditions. It controls bioluminescence in marine bacteria, ensuring that cells only produce light when enough bacteria are present to create a visible signal. It modulates virulence factor production, allowing pathogenic bacteria to delay their attack on host tissues until their numbers are sufficient to overwhelm immune defenses. It regulates conjugation, the transfer of plasmids we discussed earlier, timing genetic exchange for moments when recipient cells are plentiful. It influences competence, the ability to take up DNA from the environment. And critically, quorum sensing is at the heart of biofilm formation. As bacterial populations on a surface grow and autoinducer concentrations rise, quorum sensing triggers the expression of genes involved in producing matrix components, adhering to surfaces, and developing the three-dimensional structure of mature biofilms. The transition from planktonic to biofilm lifestyle is a coordinated, population-level decision made through this chemical communication system. This bacterial communication structure has become an exciting therapeutic target. By disrupting quorum sensing, either by degrading autoinducers, blocking their receptors, or flooding the system with artificial signals, researchers have shown they can prevent biofilm formation completely. Some experiments have demonstrated that interfering with quorum sensing can halt biofilm development even without killing bacteria, potentially delaying infectious processes long enough for immune systems to respond and clear the infection. It's an approach that sidesteps traditional antibiotic resistance mechanisms entirely, targeting bacterial coordination rather than bacterial life. Within these biofilm communities, there exists yet another layer of bacterial survival strategy, one that operates at the level of individual cells rather than populations. These are the persister cells, and they represent one of the most puzzling and clinically significant aspects of bacterial resilience. Persister cells are phenotypic variants, cells that are genetically identical to their neighbors but exist in a different physiological state. They enter a dormant, low-activity state in which they tolerate high levels of antibiotics without being resistant in the genetic sense. Unlike resistant bacteria that carry mutations or acquire genes that actively protect them from antibiotics, persisters simply shut down. They stop growing, dramatically slow their metabolism, and wait. When antibiotics arrive, they kill the actively growing, metabolically active bacteria with ease. These normal cells are fully susceptible to the drugs. But the persister cells, in their dormant state, survive. Beta-lactams that target cell wall synthesis have nothing to attack when cells aren't building walls. Aminoglycosides that kill by disrupting protein synthesis are ineffective when protein synthesis has slowed to a crawl. The antibiotics can't kill cells that aren't doing the things the antibiotics disrupt. Then, when antibiotic treatment ends and the drug clears from the system, persister cells wake up. They resume growth and reproduction, and because they're genetically identical to the original population, they remain just as susceptible to antibiotics as their ancestors were. The persistence was temporary, a phenotypic state rather than a heritable trait. But the clinical consequences are profound. Persister cells have been identified in every major bacterial pathogen and are significant contributors to chronic infections. Infections that persist despite antibiotic treatment require extended courses of antibiotics or recur repeatedly. In cystic fibrosis patients, Pseudomonas aeruginosa persisters contribute to the chronic lung infections that are so difficult to eradicate. In urinary tract infections, E. coli persisters may explain why some patients experience recurrent infections. In biofilm-associated infections on medical devices like catheters and artificial joints, persister cells within the biofilm can survive antibiotic treatment and reseed the infection once treatment stops. Research has demonstrated that persistence doesn't just enable bacteria to survive antibiotic exposure, it also serves as a pathway for the development of antibiotic resistance itself. Studies show that mutations leading to antibiotic resistance often occur after mutations that increase antibiotic tolerance. The persister state, by allowing cells to survive long enough to acquire resistance mutations, acts as an evolutionary stepping stone toward permanent resistance. The relationship between biofilms, quorum sensing, and persister cells creates a formidable defense in depth. Quorum sensing helps coordinate biofilm formation. Biofilms provide physical and metabolic protection while harboring persister cells in their depths. Persister cells survive antibiotic treatment and can repopulate the biofilm once treatment ends. Together, these mechanisms explain why certain infections, particularly those associated with implanted medical devices or damaged tissues, can be nearly impossible to cure with antibiotics alone, often requiring physical removal of infected material to achieve resolution. As we consider these community-level survival strategies, we're reminded that bacteria are not just biochemical machines responding mechanistically to their environment. They're organisms capable of sophisticated behaviors, communication, coordination, and strategic dormancy. They form societies with division of labor and collective defenses. Understanding these social aspects of bacterial life has become as important as understanding their genetics and metabolism, opening new avenues for therapeutic intervention while also deepening our appreciation for the complexity of the microbial world we're struggling to control. Among the thousands of bacterial species that exist in nature, a relatively small number have emerged as particularly dangerous adversaries in the antibiotic resistance crisis. These are not necessarily the most common bacteria or even the most virulent in an absolute sense, but they share certain characteristics that make them especially problematic. They cause severe infections, they resist multiple antibiotics, and they have spread widely in healthcare settings where vulnerable patients are most at risk. Understanding these particular pathogens, their biology, their resistance mechanisms, and their patterns of spread has become crucial to modern medicine's ability to treat life-threatening infections. The most notorious of these organisms have been grouped under an acronym that captures both their identity and their threat: ESKAPE. This stands for Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. Sometimes the acronym is extended to ESKAPE to include Escherichia coli, another major contributor to resistant infections. Together, these gram-positive and gram-negative bacteria represent the major cause of life-threatening nosocomial, or hospital-acquired, infections in immunocompromised and critically ill patients. The name ESKAPE is particularly apt because these pathogens have, in a sense, escaped the reach of commonly used antibiotics due to their increasing multi-drug resistance. They evade our best efforts to control them, slipping through gaps in our antibiotic arsenal. Recent research has demonstrated just how rapidly these organisms can develop resistance under pressure. In laboratory conditions that mimic antibiotic exposure, bacterial strains evolved resistance levels approximately 64 times higher than their ancestors within just 60 days, a mere 120 generations of bacterial reproduction. In these experiments, 87% of evolved populations reached minimum inhibitory concentrations equal to or above the peak plasma concentrations achieved in patients taking standard doses of antibiotics. In other words, within 2 months of selection pressure, most bacterial populations evolved to survive antibiotic concentrations that would be present in a treated patient's bloodstream. Among the ESKAPE pathogens, Staphylococcus aureus holds a particularly prominent place in the public consciousness, and for good reason. This gram-positive bacterium, commonly found on human skin and in the nose, is a versatile pathogen capable of causing everything from minor skin infections to life-threatening conditions like pneumonia, bloodstream infections, and endocarditis. For decades, methicillin-resistant Staphylococcus aureus, MRSA, has been synonymous with antibiotic resistance in hospitals, causing infections that are difficult and expensive to treat. But the story of MRSA has evolved in troubling ways. Initially, MRSA was almost exclusively a problem in healthcare settings, hospitals, nursing homes, dialysis centers, where it spread among patients with weakened immune systems, surgical wounds, or indwelling medical devices. We called this healthcare-associated MRSA, or HAMRSA. Then, beginning in the 1990s, a different form began to emerge: community-associated MRSA, or CAMRSA, which can be distinguished from healthcare-associated strains based on genetic, epidemiologic, and microbiological profiles. CAMRSA spreads primarily through skin-to-skin contact, contact with contaminated wounds, and in environments with poor sanitation. Transmission is facilitated by crowded living conditions, military barracks, homeless shelters, subsidized housing, correctional facilities, places where people live in close quarters with shared facilities and limited resources for hygiene. Unlike HAMRSA, which tends to affect the elderly and those with serious underlying health conditions, CAMRSA can infect young, healthy individuals with no connection to healthcare settings. The incidence of CAMRSA has risen dramatically in certain populations. In northern Saskatchewan, Canada, among Indigenous communities, the rate increased from 8.2 cases per 10,000 person-years in 2001 to 168.1 cases per 10,000 person-years in 2006. More than a 20-fold increase in just 5 years. Similar patterns have been observed among socially disadvantaged populations globally, including African Americans in the United States, Canadian First Nations communities, and Indigenous populations in Australia and New Zealand. What makes this particularly concerning is that CAMRSA strains are increasingly causing infections in hospitals. In Canada, more than 20% of nosocomial MRSA infections are now caused by CAMRSA strains. The boundary between community and healthcare has become porous, with strains flowing both directions. Both environments have become antibiotic-rich ecosystems that exchange bacterial isolates, creating a connected network of resistance that spans from homes to hospitals. One pathogen drug combination has proven especially deadly: methicillin-resistant S. aureus caused more than 100,000 deaths attributable to antimicrobial resistance in 2019 alone. The number has continued to rise, with deaths nearly doubling from 57,200 in 1990 to 130,000 in 2021. MRSA represents the single most lethal resistant pathogen drug combination we currently face. Among gram-negative pathogens, carbapenem-resistant enterobacterals, abbreviated CRE, represent one of the most serious antibiotic resistance threats. Enterobacterals is a family of bacteria that includes common pathogens like E. coli, Klebsiella pneumoniae, and Enterobacter species. Carbapenems are a class of beta-lactam antibiotics that we often hold in reserve for serious infections, particularly those caused by bacteria resistant to other drugs. The emergence and spread of carbapenem resistance therefore represents a crisis: the loss of one of our most reliable last-line antibiotics. Carbapenem resistance is primarily mediated by carbapenemase enzymes, beta-lactamases that can destroy even these powerful drugs. The most clinically important carbapenemases include Klebsiella pneumoniae carbapenemase, or KPC, and New Delhi metallo-beta-lactamase, or NDM. The story of how these enzymes have spread illustrates the global nature of antibiotic resistance. In New York City, a dramatic shift occurred in just 5 years. The annual number of NDM-positive CRE cases increased from 58 in 2019 to 388 in 2024. More significantly, NDM surpassed KPC as the most common carbapenemase type by 2024. This shift matters because infections with KPC-positive and NDM-positive CRE may require different antibiotics for effective treatment. NDM enzymes are Ambler class B metallo-beta-lactamases, meaning they use a metal ion, zinc, in their active site rather than the serine residue found in most beta-lactamases. This structural difference has important therapeutic implications. NDM enzymes are not inhibited by common beta-lactamase inhibitors like clavulanate or tazobactam, though they can be inhibited by newer inhibitors like avibactam. The global spread of NDM enzymes illustrates how quickly resistance can disseminate in our interconnected world. First identified in India in 2008, NDM genes have now been found in bacteria on every continent, spread through international travel, medical tourism, and the transfer of patients between healthcare facilities. The gene is typically carried on plasmids, allowing it to transfer between bacterial species, and it's often found alongside other resistance genes, creating bacteria resistant to virtually all available antibiotics. To understand why carbapenem resistance is so concerning, we need to appreciate what carbapenems are and why they've been so valuable. These antibiotics were originally developed from thionomycin, a natural product of Streptomyces cattleya. The first carbapenem for clinical use was imipenem, developed by Merck in the 1970s, followed by meropenem, ertapenem, and doropenem. Structurally, carbapenems are similar to penicillins. Both are beta-lactams with that characteristic four-membered ring. But carbapenems have a carbon atom instead of sulfur at a key position and possess an unsaturated bond in their five-membered ring. These structural differences make carbapenems highly resistant to most beta-lactamases that easily destroy penicillins and many cephalosporins. For decades, carbapenems worked when other beta-lactams failed. Their spectrum of activity is remarkably broad, targeting both aerobic and anaerobic bacteria, both gram-positive cocci and most gram-negative organisms. This broad activity made carbapenems ideal for treating serious infections where the identity of the pathogen wasn't immediately known, or for infections caused by multiple organisms simultaneously. The fact that they could reliably reach and kill bacteria through unusual mechanisms, like being recognized by specific porins such as the D2 porin in Pseudomonas aeruginosa due to their structural similarity to basic amino acids, gave them unique advantages. But carbapenemases have undermined this reliability. When bacteria produce these enzymes, even carbapenems are destroyed before they can reach their targets. Physicians are left with very few options, sometimes only 1 or 2 antibiotics that might still work, and sometimes none. This brings us to the polymyxins, antibiotics that include colistin and polymyxin B, discovered in the 1940s but largely set aside for decades due to serious side effects. These amphipathic molecules work by disrupting the outer membrane of gram-negative bacteria like a detergent, making cells osmotically unstable and causing death. Despite causing nephrotoxicity, kidney damage, in 30 to 60% of patients and neurotoxicity in a smaller but significant proportion, polymyxins have been reintroduced as last resort antibiotics specifically for multi-drug-resistant gram-negative bacteria including Pseudomonas aeruginosa, Klebsiella pneumoniae, Acinetobacter baumannii, and E. coli. The fact that we're using drugs with such serious side effects speaks to the desperation of the situation. When carbapenem-resistant bacteria leave us with no other options, the risk-benefit calculation shifts, and drugs that would once have been considered too toxic become justified. Recent research from 2025 has provided some reassurance. Studies have challenged conventional testing methods and shown that colistin remains effective against bacteria carrying the mobilized colistin resistance gene, called MCR, when tested under conditions that better mimic the human body, including the presence of bicarbonate and immune system components. This suggests that laboratory tests may have overestimated resistance to colistin, and that the drug may work in patients even when standard tests predict failure. Finally, we must address tuberculosis, not a member of the ESKAPE pathogens but deserving special attention as one of humanity's oldest and deadliest bacterial enemies. Mycobacterium tuberculosis, the causative agent of TB, has characteristics that make it particularly challenging to treat even before resistance enters the picture. Its waxy cell wall protects it from many antibiotics and from immune system attacks. It can persist in a dormant state within human tissues for years, hiding from both antibiotics and immune defenses. And when it does cause active disease, it requires prolonged treatment, typically 6 months of multiple antibiotics taken simultaneously. Drug-resistant tuberculosis has been classified into 5 categories by the World Health Organization, each representing a more dire situation. Multi-drug-resistant tuberculosis, or MDR-TB, is defined as TB resistant to at least isoniazid and rifampicin, the 2 most powerful first-line TB drugs. Pre-extensively drug-resistant TB, or pre-XDR-TB, is MDR-TB with additional resistance to a fluoroquinolone. Extensively drug-resistant TB, or XDR-TB, is TB resistant to rifampicin plus any fluoroquinolone plus at least one other group A drug, such as bedaquiline or linezolid. Patients with MDR-TB face higher mortality, lower rates of cure and treatment completion, higher rates of relapse, and more severe drug-related adverse effects compared to those with drug-susceptible TB. Treatment regimens are often complex and lengthy, sometimes lasting up to 2 years, with significant side effects including nausea, vomiting, hearing loss, and liver damage. The drugs are expensive, difficult to obtain in many parts of the world, and require careful monitoring to prevent serious complications. In April 2025, the WHO introduced a landmark recommendation, a novel, all-oral, 6-month regimen for treating MDR/rifampicin-resistant TB, known as BPALM/BDLLFXC. This represents significant progress, cutting treatment time from 2 years to 6 months and eliminating the need for injectable drugs that often cause hearing loss. But even this advance highlights how far behind we are with TB compared to other bacterial infections. A 6-month regimen of multiple drugs is still extraordinarily long and complex compared to treating other bacterial infections. These pathogens, the ESKAPE organisms, MRSA in its various forms, carbapenem-resistant enterobacterals, and drug-resistant tuberculosis, represent the most pressing clinical challenges in infectious disease today. They're the bacteria keeping infectious disease physicians awake at night, the organisms driving researchers to seek new antibiotics and new strategies, the reason why antibiotic stewardship has become so critical. As individuals, they're formidable. Together, they represent a threat to the foundation of modern medicine, challenging our ability to perform surgeries, care for cancer patients, manage chronic diseases, and treat routine infections. Understanding them is the first step toward developing strategies to combat them. When we speak of antibiotic resistance as a crisis, we often think first in terms of human suffering. Infections that won't heal, treatments that fail, lives cut short. These human costs are indeed at the heart of why resistance matters. But to understand the full scope of the challenge we face, we must also examine the economic dimensions of this crisis. The financial burden of antibiotic resistance ripples outward from individual patients to healthcare systems to entire economies, reshaping resource allocation and threatening to consume an ever larger share of global wealth simply trying to manage infections that were once easily treatable. In the United States, treating a bacterial infection that has become resistant to antibiotics adds nearly $1,400 to the cost compared to treating a susceptible infection. This might not sound dramatic for a single case, but consider the scale. Across the nation, the total costs attributable to antibiotic resistance exceed $2 billion annually. And that figure captures only direct healthcare costs, not the productivity losses when patients require longer hospital stays, miss more work, or suffer permanent disability. A more focused study examining Medicare populations in 2017 found that just 6 multi-drug-resistant infections contributed to $1.9 billion in healthcare costs in that single year, accounting for 11,852 deaths and 448,224 inpatient days. These 6 infections represent only a fraction of the total resistant infections occurring, yet their economic impact was already measured in billions. More recent estimates from the Centers for Disease Control indicate that treating the most alarming antimicrobial resistance threats contributes more than $4.6 billion to U.S. healthcare costs annually. The pattern of increased costs follows a predictable logic. Resistant infections require more expensive antibiotics, often newer drugs still under patent protection, or older drugs that have become scarce because they were unprofitable to manufacture. They require longer courses of treatment, sometimes weeks or months instead of days. They necessitate more laboratory testing to identify the pathogen and determine which antibiotics might still work. Patients stay in hospitals longer, occupying beds and requiring nursing care, monitoring, and supportive treatments. Complications are more common, requiring additional interventions, surgeries to remove infected tissue, intensive care for septic shock, dialysis for kidney failure caused by toxic antibiotics. When we expand our view beyond the United States to the global picture, the numbers grow staggering. Current direct healthcare costs associated with antimicrobial resistance are estimated at $66 billion per year worldwide, representing about 0.7% of global health expenditures. This is money spent managing resistant infections that, in an alternative world where resistance didn't exist, could be directed toward other health priorities. Cancer treatment, maternal and child health, chronic disease management, mental health services, health system infrastructure. But the current burden is only the beginning. Projections looking toward 2050 paint an increasingly dire economic picture if resistance rates continue following historical trends. Under business-as-usual scenarios, AMR health costs could rise to $159 billion per year by mid-century. This represents more than a doubling of current direct healthcare costs, occurring in parallel with already expected increases in healthcare spending from aging populations and advancing medical technology. These projections incorporate detailed modeling across 204 countries, analyzing 22 pathogens and 84 pathogen-drug combinations. The research examined approximately 520 million individual records spanning from 1990 to 2021, creating one of the most comprehensive pictures of resistance trends ever assembled. What emerges from this massive dataset is a clear trajectory. Without intervention, resistance will consume a growing share of healthcare resources in every region of the world. The projections become even more sobering when we consider worst-case scenarios. If resistance rates increased at the pace seen in the bottom 15% of countries, those with the weakest healthcare systems and least effective resistance control measures, AMR health costs would reach $325 billion annually by 2050. In this scenario, the global economy would be $1.7 trillion smaller in 2050 compared to baseline projections. This captures not just healthcare costs but the broader economic impact: lost productivity from sick workers, reduced economic output from premature deaths, decreased investment in regions with high infection burdens, and the drag on economic development that comes from dedicating increasing resources to healthcare rather than education, infrastructure, or innovation. The median cost of treating a resistant infection per hospital admission varies dramatically by country income level, ranging from $100 in the poorest countries to $30,000 in the wealthiest. This variation reflects differences in healthcare capacity, available antibiotics, and labor costs, but it also hints at a troubling inequity. The countries least able to afford the burden of resistant infections are often those facing the highest infection rates and the most limited treatment options. Yet these projections, sobering as they are, also contain a message of hope, or at least of opportunity. Because what the economic models also reveal is that interventions to combat antimicrobial resistance offer substantial returns on investment. This isn't simply a matter of spending more money to solve the problem. It's an observation that strategic investments in resistance control can actually reduce total healthcare spending while improving health outcomes. Consider what could be achieved through a combination of improved treatment quality and funding for innovative antibiotics. Modeling suggests these interventions could reduce health costs by $97 billion annually by 2050. Not $97 billion in new spending, but $97 billion in savings compared to the business-as-usual trajectory. At the same time, these interventions would expand the global economy by $960 billion per year and generate health benefits worth an additional $680 billion. The economic expansion comes from avoiding the productivity losses associated with severe infections, from maintaining healthcare system capacity for other conditions, and from allowing regions burdened by resistant infections to redirect resources toward economic development. Now consider a more comprehensive package: improved treatment quality, new antibiotics, enhanced vaccination programs to prevent infections before they occur, and better access to water and sanitation to reduce disease transmission. Such a package could save up to $99 billion in healthcare costs annually by 2050. The annual cost of implementing these interventions across all countries would be approximately $63 billion. Significant to be sure, but less than the healthcare savings alone, and far less than the total economic benefits. The return on investment works out to approximately 28:1 on a global scale. For every dollar spent on comprehensive AMR interventions, society gains $28 in combined healthcare savings, economic growth, and health benefits. Few public health investments offer such favorable ratios. This isn't charity. It's sound economic policy that happens to save lives and reduce suffering as a welcome side effect. The mechanisms by which these interventions generate returns are varied. Improved treatment quality means using the right antibiotics at the right doses for the right duration, maximizing effectiveness while minimizing the selective pressure that drives resistance. This requires better diagnostics to identify pathogens quickly, better access to effective antibiotics, and better trained healthcare workers who understand resistance and stewardship principles. These improvements reduce treatment failures, shorten hospital stays, and slow resistance development. Funding for innovative antibiotics means reversing the market failure that has driven pharmaceutical companies away from antibiotic development. New antibiotics with novel mechanisms can treat infections that resist existing drugs, and the availability of multiple treatment options slows resistance evolution to any single drug. Investment in new antibiotic development pays dividends for decades as those drugs enter clinical use. Enhanced vaccination programs prevent infections from occurring in the first place, eliminating the need for antibiotic treatment entirely and removing opportunities for resistance to emerge and spread. Better water and sanitation similarly reduces disease burden, particularly in low and middle-income countries where waterborne infections contribute substantially to antibiotic use. What these projections make clear is that the economic argument for addressing antimicrobial resistance is overwhelming. The cost of inaction, measured in dollars, not just lives, far exceeds the cost of comprehensive intervention. The challenge isn't finding economic justification for action, but rather mobilizing the political will and international coordination needed to implement interventions at the necessary scale. The economic burden also falls unevenly, creating justice concerns that overlay the health concerns. Low-income countries face high infection rates and limited access to effective antibiotics, creating situations where people die from infections that would be treatable in wealthy countries. Yet these same countries have the least capacity to invest in interventions, creating a vicious cycle where poverty increases infection burden, infections impede economic development, and lack of development prevents investment in infection control. Breaking this cycle requires international cooperation and resource transfer. Wealthy countries supporting interventions in poorer countries, not just for altruistic reasons but from enlightened self-interest. In our globally connected world, resistant bacteria respect no borders. A resistant strain emerging anywhere can spread everywhere through international travel and trade. An investment in containing resistance in Bangladesh or Nigeria is an investment in protecting populations in Berlin and New York. As we consider these numbers, billions in current costs, hundreds of billions in future costs, trillions in economic impact, we must remember they represent human lives and human suffering translated into economic terms. Behind each dollar of healthcare cost is a patient experiencing illness, families disrupted by serious infection, healthcare workers struggling to provide effective treatment with limited tools. The economic case for action is strong, but it reinforces rather than replaces the moral case. We have an obligation to preserve the effectiveness of antibiotics for future generations, just as we have obligations regarding climate, biodiversity, and other shared resources that transcend national boundaries and individual lifespans. Thus far, we've focused primarily on antibiotic use in human medicine, how we've deployed these drugs to treat infections, how bacteria have responded, and what this means for healthcare systems. But the story of antibiotic resistance extends far beyond hospital walls and medical clinics. To understand the full scope of how resistance develops and spreads, we must venture into agricultural fields, livestock facilities, food production systems, and the broader environment. Here we discover that the antibiotics we use to treat human illness represent only a fraction of total antibiotic consumption, and that the pathways connecting human medicine, animal agriculture, and environmental contamination form an interconnected web through which resistance flows with alarming ease. Consider this striking statistic. Worldwide, it's estimated that 70% of all antibiotics are used not in people but in farm animals. Let that proportion settle in your mind for a moment. For every antibiotic dose given to a human patient, more than 2 doses are administered to livestock. The sheer scale of agricultural antibiotic use dwarfs medical consumption, creating vast populations of bacteria exposed to these selective pressures in settings where oversight and stewardship are often minimal. The use of antibiotics in livestock production has a long history, dating back to the early 1950s when researchers discovered that adding low doses of antibiotics to animal feed promoted faster growth and improved feed efficiency. The mechanism involves suppressing gastrointestinal organisms that compete with the host animal for nutrients. By reducing these competing bacteria, antibiotics allow animals to absorb more calories from their feed, gaining weight more rapidly on less food. For producers operating on thin profit margins, this represented a substantial economic advantage. But the practice came with hidden costs that have only become apparent decades later. Much of agricultural antibiotic use has been routine and enabling, allowing animals to be kept in conditions where disease spreads easily, using antibiotics not to treat sick animals but to prevent disease in crowded, stressful environments, or simply to promote growth regardless of health status. These applications create ideal conditions for resistance development. Sustained exposure to subtherapeutic antibiotic concentrations across large populations of animals with close contact and high bacterial transmission rates. The European Union recognized these dangers and banned the use of antibiotics for growth promotion throughout its member states in 2006. According to the World Organization for Animal Health, 109 countries worldwide no longer allow growth promotion, though the practice remains permitted in at least 36 countries. Yet even where growth promotion is banned, substantial antibiotic use continues under the justification of disease prevention, a category that can be difficult to distinguish from growth promotion in practice. The geographic variation in agricultural antibiotic use is striking. In Asia and Oceania, farm antibiotic sales per livestock unit are nearly 4 times higher than in Europe. This variation reflects differences in farming practices, regulation, enforcement, and economic pressures. In regions where intensive livestock production has expanded rapidly without corresponding regulatory infrastructure, antibiotic use has grown virtually unchecked. Leading health authorities, including the European Medicines Agency and the World Health Organization, confirm that overuse of antibiotics in farming contributes to higher levels of antibiotic resistance in human infections. The mechanism isn't mysterious. Resistant bacteria that develop in farm animals can spread to humans through multiple routes. Direct contact with animals or contaminated environments for farm workers and their families. Consumption of meat, milk, or eggs containing resistant bacteria. Environmental contamination of water and soil that then exposes broader populations. And transfer of mobile genetic elements carrying resistance genes between animal-associated and human-associated bacteria. This brings us to a concern that touches every person who consumes animal products: antibiotic residues in the food supply. When animals are treated with antibiotics, those drugs and their metabolites are distributed through their tissues. Even after treatment ends, Residues can persist in meat, milk, and eggs. The primary sources of these residues are the abusive use of antibiotics in livestock, including overdosing, failure to observe withdrawal periods before slaughter or milk collection, use of contaminated water, and improper disposal of animal waste. Studies have documented antibiotic residues in food products across the globe. Research conducted in 2020 on sheep meat from Iraqi supermarkets found samples containing antibiotic residues at levels higher than maximum allowed limits. Heat treatment during cooking doesn't necessarily eliminate these residues. They can persist through normal food preparation and reach consumers in measurable quantities. The consumption of unpasteurized milk presents particular risks, as it can transmit not just antibiotic residues but also antibiotic-resistant bacteria directly to consumers. The health implications are multiple. For some individuals, antibiotic residues can trigger allergic reactions, particularly among those with known antibiotic allergies. Residues may cause gastrointestinal disturbances in sensitive individuals. Perhaps most concerning from a public health perspective, consumption of low levels of antibiotics in food may contribute to the development and maintenance of antibiotic-resistant bacteria in the human gut microbiome, creating reservoirs of resistance genes within our own bodies. For the dairy industry specifically, antibiotic residues create additional problems beyond human health concerns. Dairy yeasts and beneficial bacteria essential for manufacturing cheese, yogurt, and other fermented products are inhibited by antibiotic residues in milk, interfering with technological processes that depend on microbial fermentation. This economic impact on food production adds yet another dimension to the costs of excessive antibiotic use in agriculture. To address concerns about residues, regulatory agencies worldwide have established maximum residue limits. The maximum concentrations of veterinary drug residues that can legally remain in tissue or food products from animals that have been treated with veterinary medicines. These MRLs are designed to ensure that residue levels don't pose health hazards to consumers. The process of establishing MRLs involves careful scientific evaluation. In the European Union, the Committee for Veterinary Medicinal Products evaluates the safety of veterinary medicine residues before authorization, considering factors including acceptable daily intake, the amount that can be consumed daily over a lifetime without appreciable health risk, residue depletion studies showing how quickly drugs leave animal tissues, and analytical methods capable of detecting residues at relevant concentrations. MRLs are expressed in parts per million and vary by drug, animal species, and tissue type. Different limits apply to muscle tissue, liver, kidney, fat, milk, and eggs, reflecting the different rates at which drugs and their metabolites concentrate in various tissues. For substances considered hazardous at any level, no MRLs are established. These substances are simply prohibited from use in food-producing animals. When MRLs cannot be established for certain substances, but there's a need to monitor their presence, regulatory agencies may set reference points for action. These are technically detectable residue concentrations whose detection triggers rejection of the food consignment and investigation into how the contamination occurred. The system represents an attempt to balance the reality that we live in a chemically complex world where trace contamination is sometimes unavoidable against the need to protect public health and maintain food safety standards. Yet the problem extends beyond direct consumption of contaminated food. Approximately 60% of infectious diseases in humans are caused by zoonotic pathogens, diseases transmitted from animals to humans, and many of these pathogens can carry antibiotic resistance genes. The indiscriminate use of antibiotics in animal husbandry creates pressure on the gut microbiomes of livestock animals, selecting for resistant bacteria. Given the shorter generation times of bacteria and the high density of animals in modern farming operations, The animal gut functions as a bioreactor for breeding antibiotic-resistant bacteria. These resistant bacteria are continuously released into different environmental niches, through animal waste, during slaughter and processing, through contaminated water used in farming operations. Once in the environment, they can transfer resistance genes among native bacterial populations through the horizontal gene transfer mechanisms we've discussed. Well-documented zoonotic diseases include anthrax caused by Bacillus anthracis, bovine tuberculosis transmitted by Mycobacterium tuberculosis, brucellosis from Brucella abortus, and hemorrhagic colitis from certain strains of Escherichia coli. Each of these pathogens can carry and transmit antibiotic resistance. The World Health Organization reports that in some countries, approximately 80% of total consumption of medically important antibiotics occurs in the animal sector, largely for growth promotion in healthy animals rather than treatment of disease. The scientific evidence demonstrates conclusively that overuse in animals contributes to the emergence of antibiotic resistance that threatens human health. This isn't speculation or theoretical concern. It's documented transmission of resistant bacteria and resistance genes from agricultural settings to human populations. The environmental dimension of antibiotic resistance extends beyond agriculture to encompass the broader ecosystems through which antibiotics and resistant bacteria move. Antibiotic residues and antibiotic resistance genes are widely distributed in water and soil environments, posing ecological risks to animals and humans alike. These environmental reservoirs serve as meeting points where bacteria from different sources, human sewage, agricultural runoff, pharmaceutical manufacturing waste, can exchange genetic material and where resistance genes can persist long after the antibiotics that selected for them have degraded. Wastewater treatment plants have been identified as antimicrobial resistance hotspots. These facilities receive sewage containing antibiotics excreted by patients, bacteria shed by infected and colonized individuals, and resistance genes from countless sources. The treatment process, while removing many contaminants, doesn't completely eliminate antibiotics or resistant bacteria. Plant effluents discharge into rivers and streams, introducing antibiotic residues and resistant bacteria into aquatic ecosystems that serve as both environmental reservoirs and sources of drinking water for downstream communities. A recent study revealed an increasing prevalence of clinical antibiotic resistance genes from north to south across Europe, suggesting geographic patterns in environmental contamination that may reflect differences in antibiotic use, sewage treatment infrastructure, and environmental conditions. The use of treated wastewater for agricultural irrigation, while beneficial for water conservation, introduces additional risks by bringing antibiotic resistance genes and bacteria into contact with crops and soil ecosystems. Soil itself serves as a significant reservoir for antibiotic residues and resistance genes. Multiple inputs contribute to soil contamination. Animal manure spread as fertilizer, biosolids, treated sewage sludge applied to agricultural fields, pesticides that may contain antimicrobial compounds, and agricultural and industrial wastes. Crops irrigated with recycled water can absorb antibiotic-resistant bacteria and resistance genes, potentially posing risks to consumers who eat raw or minimally processed produce. The complexity of environmental contamination becomes apparent when we consider the sheer number of resistance genes present in a single source. More than 60 antibiotic resistance genes, including beta-lactam and tetracycline resistance genes, can be found in dairy manure alone. When this manure is spread on fields, those resistance genes enter soil ecosystems where they can persist for months or years, potentially transferring to environmental bacteria and creating long-term reservoirs of resistance. Multiple environmental compartments act as reservoirs and conduits for the spread of antibiotic-resistant bacteria. Soil exposed to contaminated water, manure, or biosolids becomes colonized by resistant bacteria. Water bodies receiving agricultural runoff or wastewater effluent carry resistant bacteria downstream. Sediments in rivers and lakes accumulate resistance genes that can be resuspended during floods or other disturbances. Air currents can carry resistant bacteria from agricultural facilities to neighboring communities. The environment isn't merely a passive recipient of resistance genes, but an active participant in their dissemination and evolution. Human activity can contaminate the environment with antibiotics and antifungals, speeding up the development and spread of antimicrobial resistance through several mechanisms. Antibiotics in the environment exert selective pressure on environmental bacteria, favoring those carrying resistance genes. Subinhibitory concentrations, too low to kill bacteria but high enough to exert selection, are particularly problematic because they promote resistance without eliminating susceptible bacteria, maintaining diverse populations while shifting the balance toward resistance. Environmental bacteria can serve as a reservoir of resistance genes that can later transfer to pathogenic bacteria affecting humans and animals. The One Health perspective, which recognizes the interconnected health of humans, animals, and the environment, emerges as essential for understanding and addressing antibiotic resistance. Resistance developing in livestock doesn't stay in livestock. Resistance genes in environmental bacteria don't remain permanently environmental. The boundaries are porous, and the flows are bidirectional. Bacteria and genes move from humans to animals, from animals to the environment, from the environment back to humans. Interventions focus solely on human medicine while ignoring agricultural and environmental dimensions will be incomplete at best and ineffective at worst. As we consider these pathways, antibiotics administered to livestock, residues persisting in food, resistant bacteria spreading from farms, environmental contamination through multiple routes, we begin to appreciate that antibiotic resistance is truly a systems-level problem. It cannot be solved through medical interventions alone. It requires coordinated action across sectors, stricter regulation of agricultural antibiotic use, improved animal husbandry practices that reduce disease pressure without relying on routine antibiotic administration, enhanced wastewater treatment to remove antibiotics and resistant bacteria before discharge, agricultural practices that minimize contamination of soil and water, and global cooperation to raise standards in regions where oversight is currently minimal. The challenge is daunting, but understanding the full scope of the problem is the first step toward developing comprehensive solutions. As we've explored the mechanisms of resistance and the pathways through which it spreads, a question naturally arises. Is antibiotic resistance permanent? Once bacteria evolve resistance, are they forever changed? Or might resistance fade if we simply stopped using the antibiotics that's selected for it? To answer these questions, we must delve into the evolutionary biology of resistance. Understanding not just how resistance emerges, but how it persists, what costs it imposes on bacteria, and under what conditions it might potentially be reversed. The story begins with a concept that seems almost intuitive. Resistance should come at a price. When bacteria acquire mutations that confer antibiotic resistance, or when they take up and maintain resistance genes on plasmids, they're allocating cellular resources to these defensive mechanisms. They're producing enzymes that degrade antibiotics, operating efflux pumps that require energy to run, maintaining altered target molecules that may not function quite as efficiently as the originals, or carrying extra genetic material that must be replicated with each cell division. All of these should impose what evolutionary biologists call fitness costs, reductions in the bacteria's ability to grow, reproduce, or compete when antibiotics aren't present. Meta-analyses examining fitness costs across multiple studies have confirmed this intuition. On average, antibiotic-resistant mutants show decreased biological fitness compared to susceptible strains. The mean relative fitness of resistance mutations across diverse studies is approximately 0.90, indicating that resistant bacteria grow about 10% more slowly or reproduce slightly less efficiently than their susceptible counterparts in antibiotic-free environments. This might not sound like much, but in the competitive world of bacteria, where slight growth advantages can determine which strains dominate a population, a 10% fitness difference is substantial. However, and this is where the story becomes more complex, the magnitude of fitness costs varies tremendously depending on the bacterium, the specific resistance mechanism, and the environmental context. Gram-positive bacteria show significantly greater fitness costs, with mean fitness around 0.822, compared to gram-negative bacteria at 0.973. This suggests that the same resistance mechanisms exact different tolls depending on bacterial biology. The relative fitness of resistance mutations is also negatively correlated with the fold increase in minimum inhibitory concentration. The more resistant a bacterium becomes, the greater the fitness cost it typically pays. The fold increase in mic accounts for nearly 23% of the variation in fitness costs, a substantial portion but far from the whole story. If resistance mutations impose fitness costs, simple evolutionary logic suggests that when antibiotics are removed, susceptible bacteria should outcompete resistant ones. The resistant bacteria, burdened by the costs of maintaining unnecessary defensive mechanisms, should gradually decline in frequency as fitter susceptible bacteria reproduce more successfully. Over time, we might expect bacterial populations to revert to susceptibility, solving the resistance problem simply by withdrawing the selective pressure that caused it. This reasoning isn't entirely wrong, but it's incomplete. The magnitude of fitness costs required for reversion determines how long it would take to reduce the abundance of resistant bacteria, and that time is inversely related to the cost of resistance. Small fitness costs mean slow reversion, potentially taking years or decades even in the complete absence of antibiotic use. During those long periods, resistant bacteria continue circulating, causing infections, and potentially spreading their resistance genes to other bacteria. Moreover, two major mechanisms can prevent resistance reversal even when antibiotics are removed entirely. The first is genetic linkage between resistance genes and other selected loci. If a resistance gene happens to be physically close to another gene that provides a fitness advantage in antibiotic-free environments, a phenomenon called genetic hitchhiking, the resistance gene can be maintained in the population simply because bacteria carrying both genes do well, even if the resistance gene itself is neutral or slightly deleterious. This genetic co-selection means resistance genes can persist for reasons unrelated to antibiotic pressure. The second and more important mechanism is compensatory evolution, the acquisition of secondary mutations that restore fitness without eliminating resistance. This phenomenon fundamentally changes the evolutionary dynamics of resistance and explains why simply stopping antibiotic use is rarely sufficient to reverse resistance in bacterial populations. Here's how compensatory evolution works. A bacterium acquires a resistance mutation that slows its growth, perhaps by altering a ribosomal protein that makes protein synthesis slightly less efficient, or by producing a modified enzyme that provides resistance but doesn't perform its normal cellular function quite as well. In an environment without antibiotics, this bacterium is at a competitive disadvantage. But now imagine a second mutation occurs elsewhere in the genome, perhaps in a regulatory gene or in a protein that interacts with the altered target. This second mutation doesn't reverse the resistance. The bacterium remains just as resistant to antibiotics as before. But it does compensate for the fitness cost, restoring growth rate or competitive ability to levels approaching or matching the original susceptible strain. Once compensatory mutations arise, the fitness disadvantage that should have driven resistant bacteria out of the population disappears. The bacteria are now both resistant and fit. Removing antibiotics from the environment no longer creates selective pressure for reversion because the resistant bacteria compete effectively with susceptible ones. The resistance has been stabilized. Research on double-resistant Escherichia coli strains, bacteria carrying mutations conferring resistance to both rifampicin and streptomycin, demonstrates how this process works in detail. These double resistance mutations create large fitness costs because of epistatic interactions. The combined effect of having both resistance mutations is worse than the sum of their individual effects. The bacteria grow poorly and compete badly against susceptible strains. But compensatory evolution opens alternative evolutionary paths for rapid fitness recovery. Mutations in genes like MAR, a transcriptional regulator, and ENVZ, involved in osmoregulation, were found to increase fitness specifically in antibiotic-resistant genotypes while being neutral or even deleterious in wild-type strains. These mutations compensated for the epistasis between resistance alleles. They specifically ameliorated the fitness costs that arose from having both resistance mutations together. Crucially, compensatory mutations themselves often carry fitness costs. They improve fitness relative to the uncompensated resistant strain, but they don't necessarily restore fitness all the way back to the level of the original susceptible strain. This prevents antibiotic-resistant bacteria from completely matching the fitness of sensitive variants, maintaining some residual cost of resistance. However, the existence of compensatory mutations makes genetic reversion of resistance far less likely than it would otherwise be, because the fitness pressure driving reversion is substantially reduced. The broader range of genetic targets for compensation compared to genetic reversion is also significant. To reverse resistance through mutation, bacteria typically need to revert the exact mutation that caused resistance, a very specific genetic change that may occur rarely. But to achieve compensation, mutations in many different genes might suffice, as long as they somehow ameliorate the resistance-associated fitness costs. The larger mutational target makes compensatory evolution much more probable than true reversion. Research on multi-drug-resistant bacteria has shown that compensatory evolution can stabilize costly multiple resistances. When bacteria carry resistance to multiple antibiotics simultaneously, the combined fitness costs can be severe. Yet through compensatory mutations, even these heavily burdened bacteria can recover fitness. Studies have documented rapid compensatory evolution in bacteria with resistance to several drugs, with secondary mutations emerging that specifically address the fitness costs imposed by multiple resistance determinants. The same principles apply to plasmid-mediated resistance. Common conjugal plasmids carrying resistance genes often impose fitness burdens on their bacterial hosts. The cost of replicating the extra DNA, expressing plasmid genes, and maintaining the plasmid through cell divisions. Yet these plasmids can be maintained in bacterial populations via horizontal transfer even in the absence of antibiotic selection. The ability to transfer between cells creates opportunities for plasmids to spread faster than they're lost, maintaining them in populations despite fitness costs. But compensatory evolution adds another layer of stability. Bacteria can rapidly evolve chromosomal mutations that ameliorate the fitness burden imposed by resistance plasmids without losing the plasmid or the resistance genes it carries. The bacteria adapt to carrying the plasmid, reducing its cost while retaining its benefits. Studies have shown that under sustained growth in antibiotic-free environments without opportunities for horizontal transfer, conditions specifically designed to prevent plasmid reacquisition. Plasmids imposing fitness costs can eventually be lost from populations. But these conditions rarely exist in nature, where horizontal transfer continues and where compensatory evolution stabilizes plasmid carriage. The possibility of reversal exists under specific, constrained conditions. When plasmid-carrying bacteria face sustained growth without antibiotics and without opportunities for conjugation, and when compensatory mutations haven't yet ameliorated plasmid costs, selection can drive plasmid loss. The rate depends critically on the magnitude of the fitness burden and whether compensation has occurred. But in most real-world settings, where horizontal transfer continues, where antibiotic use hasn't completely ceased, and where bacteria have had time to evolve compensatory mutations, plasmid-mediated resistance persists stubbornly. Clinical evidence on the effectiveness of stopping antibiotic treatment to reduce resistance presents a mixed picture, and this variation makes sense in light of what we now understand about fitness costs and compensation. In some cases, reducing antibiotic use has corresponded with decreases in resistance frequencies, suggesting that fitness costs can drive reversion under certain circumstances. In other cases, resistance has persisted or even increased despite reduced antibiotic use, consistent with compensatory evolution having stabilized the resistant populations. A crucial insight emerges when we compare laboratory evolution studies with clinical observations. There are limits to compensatory adaptation in nature that laboratory studies don't fully capture. Numerous laboratory experiments demonstrate that antibiotic resistance can be stably maintained in the absence of antibiotics through compensatory evolution. Yet clinical studies generally show that resistance in pathogen populations declines after antibiotic use is stopped. This apparent disagreement reflects important differences between laboratory conditions and natural settings. First, clinical antibiotic treatment typically fails to completely eradicate antibiotic-sensitive strains. Reservoirs of susceptible bacteria persist in patients who haven't been treated, in body sites where antibiotics don't penetrate well, and in environmental sources. When antibiotic use decreases, these susceptible strains can recolonize and compete with resistant strains. In the presence of this competition, even small residual fitness costs associated with resistance, costs that remain after partial compensation, can tip the balance towards susceptible strains. Second, laboratory studies may overestimate the efficacy of compensatory adaptation by failing to capture fitness costs associated with the compensatory mutations themselves. In carefully controlled laboratory conditions with simple environments and single stressors, compensatory mutations might fully restore fitness. But in the complex, variable environments that bacteria face in nature, with fluctuating temperatures, nutrient availabilities, pH levels, immune pressures, and competition from diverse microorganisms, compensatory mutations may have pleiotropic effects, improving fitness in some contexts while reducing it in others. Third, laboratory experiments typically use clonal populations or populations with limited genetic diversity, while natural pathogen populations are genetically heterogeneous. In diverse populations, different lineages compete not just based on resistance versus susceptibility but based on numerous other fitness-affecting traits. The dynamics of resistance maintenance become more complex when embedded in this broader competitive landscape. These factors explain why selection for compensatory adaptation may not stabilize resistance in natural populations of pathogenic bacteria to the same extent observed in laboratory conditions. The implication is encouraging. Reducing antibiotic use can indeed reduce resistance prevalence in clinical settings, though the process may be slower than we'd like and may require sustained reductions maintained over years or decades. Beyond fitness costs and compensation, there exist additional barriers that can limit the spread of resistance genes through horizontal transfer. When resistance genes move between bacterial species or strains, they enter new genetic and cellular contexts. The incoming gene may not work well in its new host due to fundamental incompatibilities, barriers that exist at the phenotypic and genetic levels. Research examining 6 different ribosomal protection protein genes expressed in Escherichia coli revealed highly variable outcomes that illustrate these barriers. 2 of the genes, TET-M and TET-Q, conferred high-level tetracycline resistance without reducing bacterial fitness. These genes transferred successfully, maintaining their function and imposing minimal cost. 2 others, TET-O and TET-W, conferred high-level resistance but significantly reduced growth fitness due to suboptimal codon usage patterns. The genes functioned, providing resistance, but because they used codons that were rare in E. coli, the bacteria struggled to produce enough of the corresponding transfer RNAs to translate these genes efficiently. The result was a codon usage incompatibility that created a fitness burden. Another gene, TET-B-P, conferred only low-level resistance and experienced transcriptional silencing associated with its unusual nucleotide composition. The gene was present but not expressed at sufficient levels because its sequence characteristics didn't match the host's preferences for gene regulation. Finally, ATR-A didn't confer resistance at all in the new host, and resistant mutants couldn't be selected. This gene, despite functioning in its original host, completely failed in E. coli. Evolution experiments demonstrated that codon usage incompatibility and transcriptional regulation defects create substantial barriers to successful establishment of horizontally transferred resistance genes. Bacteria receiving genes with poor codon usage would need to evolve changes in their transfer RNA pools or mutations in the transferred gene to use more common codons. Evolutionary adjustments that take time and may never occur if the initial fitness costs are too severe. These findings explain why, despite the existence of at least 12 distinct ribosomal protection protein classes, TET-M remains the predominant variant in E. coli while other classes face obstacles to fixation. Not all resistance genes are equally transferable. Some spread easily because they work well in many different hosts. Others are confined to narrow host ranges because of these genetic and phenotypic barriers. This variation in transferability has implications for resistance spread. Resistance mechanisms that work well across diverse bacterial species, those without significant codon usage problems, without unusual regulatory requirements, and without complex dependencies on host cell machinery, will spread more readily through horizontal transfer. Conversely, resistance mechanisms that impose barriers will remain more confined, spreading primarily through vertical inheritance within lineages that have already adapted to carrying them. As we step back from these detailed evolutionary considerations, a picture emerges of resistance as a dynamic, complex phenomenon. Resistance can arise rapidly through mutation and horizontal transfer. Once established, it can be stabilized through compensatory evolution, making it difficult to eliminate even when antibiotics are removed. Yet barriers exist: fitness costs that have not been fully compensated, genetic incompatibilities that limit horizontal transfer, competition from susceptible strains when selection pressure is removed. These barriers mean resistance is not inevitable or irreversible, but overcoming resistance requires sustained, coordinated efforts maintained over long time periods. Understanding the evolutionary dynamics doesn't just satisfy scientific curiosity. It informs strategies for resistance management, helping us understand when interventions might succeed and what forms those interventions should take. Having explored the grand sweep of resistance evolution, its mechanisms, its costs, its potential reversibility, we now turn our attention to a more practical question. How do physicians actually determine which antibiotics will work for a particular infection, and how do they optimize treatment to maximize effectiveness while minimizing the emergence of resistance? The answers involve sophisticated laboratory testing, mathematical modeling of drug behavior in the body, and careful monitoring of both therapeutic effects and potential toxicity. These clinical tools represent medicine's attempt to bring precision to antibiotic therapy, transforming what was once largely empirical, guided by experience and educated guessing, into something approaching a science. At the foundation of antibiotic testing lies a simple but crucial measurement: the minimum inhibitory concentration, universally abbreviated as MIC. This is defined as the lowest concentration of an antibiotic that prevents visible growth of a bacterium in laboratory culture. The test involves exposing bacteria to a series of antibiotic concentrations, perhaps starting at very low levels and doubling with each step, and observing which concentration first stops the bacteria from growing. MIC testing is performed in both diagnostic laboratories, where it guides treatment decisions for individual patients, and in drug discovery laboratories, where it helps identify promising new antibiotic candidates. The results are expressed in micrograms per milliliter or milligrams per liter, standardized units that allow meaningful comparisons across different antibiotics, different bacteria, and different laboratories. But a raw MIC value alone doesn't tell a physician whether a particular antibiotic will successfully treat an infection. That number must be interpreted in clinical context, which is where breakpoints come in. Clinical breakpoints are agreed-upon threshold values that classify bacteria as susceptible, intermediate, or resistant to a particular antibiotic based on their MIC. These breakpoints are published by standards development organizations, including the U.S. Clinical and Laboratory Standards Institute, the British Society for Antimicrobial Chemotherapy, and the European Committee on Antimicrobial Susceptibility Testing. The process of establishing breakpoints considers multiple factors. First, what concentrations of the antibiotic can be safely achieved in patients taking standard doses? This depends on the drug's pharmacokinetics, how it's absorbed, distributed, metabolized, and eliminated. Second, where in the body is the infection located? Antibiotics reach different concentrations in blood, urine, cerebrospinal fluid, and tissues, so a concentration sufficient for treating a urinary tract infection might be inadequate for treating meningitis. Third, what is the relationship between antibiotic concentration and microbiological effect? At what concentrations does bacterial killing occur reliably? Integrating these considerations, breakpoints denote MIC values below which an infection is likely to be highly treatable. A bacterium with an MIC well below the breakpoint, meaning it's inhibited by very low antibiotic concentrations, should be readily killed by standard treatment. A bacterium with an MIC near or above the breakpoint may require higher doses, longer treatment courses, or alternative antibiotics. The principle underlying breakpoints is straightforward. The lower the MIC, the more potent the antimicrobial. The further the MIC is from the breakpoint, the more susceptible the organism and the more confident we can be that standard therapy will succeed. Conversely, when MICs approach or exceed breakpoints, treatment becomes uncertain, and adjustments may be necessary. But determining the right dose to prescribe involves more than just knowing the MIC and whether it falls above or below a breakpoint. This is where pharmacokinetic pharmacodynamic modeling enters the picture, a sophisticated framework for quantitatively linking drug exposure to microbiological and clinical outcomes. The name is lengthy, but the concept is logical. Pharmacokinetics describes what the body does to the drug, while pharmacodynamics describes what the drug does to the microbe. Pharmacokinetics encompasses absorption from the site of administration, distribution throughout body tissues, metabolism by liver enzymes or other pathways, and elimination through kidneys, bile, or other routes. These processes determine the concentration time profile, how drug levels rise and fall in blood and tissues over the course of treatment. Different drugs show dramatically different profiles. Some are absorbed rapidly and eliminated quickly, requiring frequent dosing. Others persist for long periods, allowing once daily or even less frequent administration. Pharmacodynamics characterizes the relationship between drug concentration and microbial response. For some antibiotics, killing depends primarily on achieving high peak concentrations. The higher the peak, the more bacteria are killed, following what's called concentration-dependent killing. For others, what matters most is the duration of time that drug concentrations remain above the mic. These show time-dependent killing. Still others show an intermediate pattern where both peak concentration and duration above mic contribute to efficacy. Three key pharmacokinetic pharmacodynamic indices have emerged as predictors of efficacy. The first is the ratio of maximum drug concentration to minimum inhibitory concentration, written as Cmax/mic. This index matters most for concentration-dependent antibiotics like aminoglycosides and fluoroquinolones. A higher ratio means more effective killing, and clinical studies have established target ratios associated with good outcomes. For gentamicin treatment of bloodstream infections, Cmax/mic ratios of 8 or higher correlate with significantly improved outcomes. The second index is the time that drug concentration remains above the mic, written as time-mic. This matters most for time-dependent antibiotics like beta-lactams. For these drugs, keeping concentrations above the mic for a certain percentage of the dosing interval, often 40 to 70% depending on the specific drug and infection, predicts success. Achieving higher peak concentrations beyond what's needed to exceed the mic provides little additional benefit. What matters is sustained exposure above the threshold. The third index is the area under the concentration time curve divided by mic, written as AUC/mic. This captures total drug exposure relative to bacterial susceptibility and predicts efficacy for antibiotics like fluoroquinolones and aminoglycosides. For gentamicin, AUC/mic ratios exceeding 70 correlate with therapeutic success in treating bacteremia. By integrating pharmacokinetic and pharmacodynamic information, clinicians can predict how different dosing regimens will influence antimicrobial activity. They can determine whether a drug will achieve concentrations exceeding the mic at the infection site, whether those concentrations will be sustained for adequate duration, and whether the resulting bacterial killing will be sufficient for clinical cure. This framework has proven especially critical for treating critically ill patients whose altered physiology, increased fluid volumes, enhanced kidney clearance, reduced absorption, can dramatically change drug concentrations compared to healthy volunteers. Standard dosing strategies developed for stable patients are unlikely to consistently achieve target pharmacokinetic pharmacodynamic parameters in ICU patients, necessitating individualized dose adjustments guided by PK/PD principles. For certain antibiotics, the margin between therapeutic and toxic concentrations is narrow enough that therapeutic drug monitoring becomes necessary. These narrow therapeutic index drugs require careful tracking of blood levels to ensure efficacy without causing harm. Among antibiotics, aminoglycosides and vancomycin are the classic examples requiring monitoring, while most other antibiotic classes have wide enough therapeutic windows that monitoring is unnecessary. The rationale for monitoring aminoglycosides reflects their pharmacodynamic characteristics and toxicity profile. Bactericidal efficacy is directly related to peak concentrations. Higher peaks mean better killing through concentration-dependent mechanisms. But toxicity, including kidney damage that is usually reversible and hearing loss that is often permanent, relates to total drug exposure over time. The goal is achieving high peaks for efficacy while limiting overall exposure to reduce toxicity risk. This requires a delicate balance that varies among patients based on kidney function, body composition, and other factors. Vancomycin similarly has a low therapeutic index, with both kidney damage and hearing loss complicating therapy. The drug also shows considerable variability in pharmacokinetics between patients. Two patients receiving the same dose may achieve very different blood concentrations. Without monitoring, some patients receive too little drug, risking treatment failure and resistance development, while others receive too much, risking toxicity. The need for monitoring relates fundamentally to inherent variability in pharmacokinetic and pharmacodynamic measures. For narrow therapeutic index drugs, small changes in drug concentration, whether from differences in absorption, distribution, metabolism, or elimination, can shift outcomes from therapeutic to subtherapeutic or toxic. Monitoring allows clinicians to measure actual concentrations achieved in individual patients and adjust doses accordingly, maintaining concentrations within the therapeutic range. Most antibiotics don't require this level of monitoring because their therapeutic indices are wide. Beta-lactams, macrolides, and fluoroquinolones, for instance, can be given across a broad dose range with good efficacy and acceptable toxicity. The standard doses work well for most patients, and the safety margins are sufficient that occasional deviations from target concentrations don't cause problems. But for the minority of antibiotics with narrow indices, monitoring represents an essential tool for optimizing the risk-benefit balance. The importance of rapid and accurate diagnosis has led to the development of faster methods for identifying pathogens and determining their antibiotic susceptibility. Conventional approaches require growing bacteria from clinical specimens on solid media, identifying them to species level through biochemical tests or mass spectrometry, and then performing susceptibility testing, a process requiring 48 to 72 hours from initial specimen collection to final results. For bloodstream infections, where every hour of delay in appropriate therapy increases mortality risk, this timeline is unacceptably long. Rapid diagnostic methods are revolutionizing management of bacterial infections by compressing this timeline dramatically. The European Committee on Antimicrobial Susceptibility Testing developed a standardized rapid antibiotic susceptibility testing method, RAST, that provides results at 4, 6, and 8 hours directly from positive blood culture bottles for key pathogens including E. coli, K. pneumoniae, P. aeruginosa, and S. aureus. Rather than waiting for overnight growth on solid media, this method tests bacteria directly from blood culture bottles, taking advantage of the fact that enough bacterial growth has already occurred to produce a positive blood culture. Even more advanced methods combine single-cell phenotypic susceptibility testing with fluorescence in situ hybridization in microfluidic devices, sophisticated instruments that manipulate tiny volumes of fluids in microscopic channels. These systems can provide antibiotic susceptibility test results in as little as 30 minutes to 2 hours for urine samples. Some achieve 100% sensitivity for both pathogen identification and susceptibility determination in samples containing multiple bacterial species, a common situation in urinary tract infections where contamination with skin flora can complicate interpretation. The clinical impact of rapid diagnostics extends beyond faster results. Knowing within hours rather than days which antibiotics will work allows physicians to narrow therapy quickly, moving from broad-spectrum empirical coverage, which affects many bacterial species and creates strong selection for resistance, to narrow-spectrum targeted therapy against a specific pathogen causing infection. This narrowing, often called de-escalation, reduces unnecessary antibiotic exposure, lessens disruption to beneficial microbiota, decreases costs, and may slow resistance development. One aspect of antibiotic classification that physicians have traditionally considered when selecting therapy is whether drugs are bactericidal, actively killing bacteria, or bacteriostatic, halting bacterial growth without necessarily killing microorganisms. Bactericidal agents include beta-lactams, aminoglycosides, and fluoroquinolones, which disrupt vital cellular processes or damage structures beyond repair. Bacteriostatic agents include tetracyclines, macrolides, and linezolid, which halt growth and allow the immune system time to eliminate pathogens. Traditional medical teaching held that bactericidal agents were superior to bacteriostatic agents, especially in severe infections like endocarditis, in patients with compromised immune systems such as those with neutropenia, and in bloodstream infections. The reasoning seems sound. If bacteria are actively killed rather than merely inhibited, infections should resolve faster and more reliably. Traditional teaching also warned against combining bactericidal and bacteriostatic antibiotics, fearing antagonism, that the bacteriostatic drug might interfere with the bactericidal drug's ability to kill actively growing bacteria. However, comprehensive clinical evidence has challenged this dogma substantially. A meta-analysis published in 2015 examined 33 randomized controlled trials comparing bacteriostatic with bactericidal antibiotics in severe infections. The analysis found no significant differences in clinical cure rates, mortality rates, or relapse rates between the two classes. Patients treated with bacteriostatic antibiotics did just as well as those treated with bactericidal agents. Subgroup analysis focusing specifically on 13 pneumonia studies confirmed these findings. No significant difference between bactericidal and bacteriostatic antibiotics in treating one of the most common and serious bacterial infections. Specific bacteriostatic agents, including linezolid and tigecycline, have proven clinically non-inferior to bactericidal agents in multiple infection types: pneumonia, intra-abdominal infections, and skin and soft tissue infections. In head-to-head comparisons, patients receiving bacteriostatic drugs achieved cure rates and survival comparable to those receiving bactericidal agents. These findings suggest that the bactericidal-bacteriostatic distinction, while valid at the microbiological level, the drugs truly do differ in their mechanisms, may be less clinically important than traditionally believed. In patients with functioning immune systems, bacteriostatic drugs that halt bacterial growth allow immune cells to catch up and eliminate the infection. The outcome is similar whether bacteria are killed by antibiotics or by the immune system once the drugs stop bacterial proliferation. The challenge to traditional teaching extends to the question of combination therapy. While specific combinations should still be avoided based on known antagonism, and some infections like endocarditis do require bactericidal therapy based on clinical evidence, the blanket prohibition against combining bactericidal and bacteriostatic antibiotics appears less justified than once thought. Each combination and each clinical scenario should be evaluated based on evidence rather than assumed incompatibility based on mechanism classification. As we synthesize these clinical tools, mic testing and breakpoints guiding drug selection, PK/PD modeling optimizing dosing, therapeutic drug monitoring maintaining concentrations in the therapeutic range, rapid diagnostics accelerating appropriate therapy, and evidence-based rather than dogma-based understanding of bactericidal versus bacteriostatic effects. We see medicines attempt to bring rigor and precision to antibiotic use. These aren't merely academic exercises but practical tools that improve outcomes, reduce toxicity, and potentially slow resistance development by ensuring that when we use antibiotics, we use them as effectively as possible. The complexity of these approaches reflects the high stakes involved. When antibiotics fail, patients die, and when we use antibiotics poorly, we hasten the day when they fail more often. Throughout our journey, we've encountered bacteria's remarkable ability to evolve resistance to individual antibiotics. A natural question arises: what if we don't give them that opportunity? What if we attack bacteria with multiple antibiotics simultaneously, forcing them to evolve resistance to several drugs at once? This is the fundamental logic behind combination antibiotic therapy, and it represents one of medicine's strategies for staying ahead in the evolutionary arms race with bacterial pathogens. The theoretical foundation is compelling. When bacteria face a single antibiotic, a mutation that confers resistance to that drug provides a survival advantage. Those resistant mutants proliferate, eventually dominating the population. But when bacteria face two antibiotics simultaneously, resistance requires acquiring mutations for both drugs. One mutation alone is insufficient for survival. The probability of a single bacterial cell spontaneously acquiring both resistance mutations is the product of the individual mutation rates, which can be vanishingly small. If resistance to drug A occurs in 1 in 10 million cells, and resistance to drug B occurs in 1 in 10 million cells, then resistance to both simultaneously would be expected in only 1 in 100 trillion cells, a number that may exceed the entire bacterial population in a localized infection. This genetic barrier to resistance represents combination therapy's primary advantage for limiting resistance evolution. Research demonstrates that combination therapy is almost always superior to monotherapy in preventing resistance development, even when the total drug dose is held constant. It's not simply about using more antibiotic. It's about the synergistic effect of requiring multiple independent adaptations simultaneously. Successful examples of this approach are well documented. The treatment of tuberculosis, which we discussed earlier, has relied on combination therapy since the 1950s. Patients receive 3 or 4 antibiotics simultaneously for months, specifically to prevent the emergence of resistance. HIV treatment follows similar principles, using combinations of antiretroviral drugs to suppress viral evolution. These success stories demonstrate that when the barriers to resistance are made high enough through intelligent combination of drugs, even rapidly evolving pathogens can be controlled. Beyond preventing resistance, combinations can sometimes rejuvenate the effectiveness of old antibiotics to which bacteria had previously developed resistance. In some cases, combining two drugs creates synergistic effects where their joint activity exceeds the sum of their individual activities, achieving better bacterial killing than either drug alone. Combinations have successfully eliminated multi-drug resistant and biofilm-forming organisms with significantly lower rates of resistance development and reduced side effects compared to high-dose monotherapy. But the concept of synergy, that special interaction where combined drugs produce effects greater than the sum of their parts, requires careful definition and even more careful interpretation. In laboratory settings, synergy is typically defined by specific mathematical criteria. If drug A alone inhibits bacterial growth by 50% and drug B alone also inhibits by 50%, we might expect the combination to inhibit by 100%. A simple additive effect. Synergy occurs when the combination inhibits more than 100% in this example, achieving greater bacterial killing than predicted from the individual activities. Historical studies from the 1980s examining combinations of beta-lactam and aminoglycoside antibiotics demonstrated that such synergistic activity could be achieved against gram-negative bacteria. The rationale seemed sound. Beta-lactams disrupt cell wall synthesis, potentially allowing aminoglycosides to penetrate more easily and reach their ribosomal targets. In laboratory assays, combinations with amikacin showed inhibitory synergistic activity in 29% of tests against gentamicin-resistant isolates. The most active combination tested, amikacin with ceftriaxone, demonstrated synergism in 67% of assays. These laboratory findings led to widespread clinical use of beta-lactam and aminoglycoside combinations, particularly for serious gram-negative infections. The approach became standard practice in many settings, with the expectation that synergy observed in test tubes would translate to better patient outcomes. But more recent research has revealed a more nuanced and sometimes troubling picture. A comprehensive study published in 2022 examined 254 E. coli bloodstream infection strains, testing 4 different beta-lactam and aminoglycoside combinations. While most strain drug combinations showed additive interactions, the drugs worked together as expected without antagonism. No strains demonstrated synergistic responses. Even more unexpectedly, approximately 3% of strain drug combinations exhibited clinically relevant antagonism, where the combination performed worse than single drugs alone. The interaction profiles varied significantly across bacterial strains, revealing that synergy is highly strain-specific rather than universal. The same antibiotic combination that shows additivity in one bacterial isolate might show antagonism in another, genetically similar isolate. This strain-to-strain variation means that laboratory testing on reference strains may not predict clinical behavior against the diverse pathogens causing infections in patients. Perhaps most significantly, studies comparing combination therapy versus beta-lactam monotherapy found no association between combination use and beneficial effects in preventing resistance development. The theoretical advantage of combination therapy, raising the genetic barrier to resistance, didn't materialize in clinical practice for these particular combinations. This raises important questions about the routine use of beta-lactam and aminoglycoside combinations, particularly given that aminoglycosides carry significant toxicity risks, including kidney damage and hearing loss. The disconnect between laboratory synergy and clinical outcomes highlights a fundamental challenge in antibiotic pharmacology. What happens in a test tube, where bacterial cells are exposed to constant antibiotic concentrations in simple growth media, may not reflect what happens in patients, where drug concentrations fluctuate over time. Bacteria exist in complex tissue environments with variable drug penetration, and host immune responses contribute substantially to bacterial clearance. In vitro synergy doesn't guarantee in vivo synergy, and even demonstrated in vivo synergy in animal models doesn't always predict improved outcomes in human patients. Despite these sobering findings for some combinations, other evidence suggests that properly designed combinations offer real benefits. Some studies do link antibiotic synergy to better clinical outcomes, though the correlations are inconsistent and the benefits modest. The key seems to be moving beyond empirical combination use, throwing 2 drugs together and hoping for synergy, toward rationally designed combinations based on understanding of bacterial physiology and resistance mechanisms. This brings us to an emerging strategy: developing antibiotics that inherently target 2 distinct bacterial processes within a single molecule. Rather than combining 2 separate drugs and hoping they work synergistically, researchers are designing dual-targeting antibiotics, single chemical compounds engineered to inhibit 2 different bacterial targets simultaneously. Research on compounds like SCH, 79797 demonstrates the potential of this approach. This compound targets both dihydrofolate reductase, an enzyme involved in folate metabolism, and the bacterial membrane itself, creating damage through two completely independent mechanisms. In laboratory evolution experiments conducted over 25 days, researchers successfully isolated mutants resistant to control antibiotics like novabiocin, trimethoprim, and nisin. But no SCH 79797 resistant mutants emerged. The compound's dual mechanism created a resistance barrier that bacteria apparently couldn't overcome within the experimental time frame. A 2025 study focusing specifically on dual-target permeabilizers, antibiotics that simultaneously disrupt membrane integrity and block another cellular pathway, demonstrated that this class shows limited resistance development in ESKAPE pathogens. The research hypothesized that dual targeting alone is insufficient. Rather, only antibiotics that combine membrane disruption with inhibition of another cellular target display genuinely reduced resistance development. The logic is elegant. Mutations that increase membrane stability to resist membrane-disrupting effects might make cells more vulnerable to the second mechanism. Mutations that confer resistance to the second mechanism might compromise membrane function, increasing vulnerability to membrane disruption. The two resistance mechanisms work at cross purposes, creating evolutionary trade-offs that prevent effective resistance. The probability of simultaneously acquiring resistance to both independent targets is extraordinarily low, the product of individual mutation rates, just as with combination therapy, but now within a single drug that ensures both targets are always hit together at the right ratio. This concept of evolutionary trade-offs leads us to another fascinating strategy: collateral sensitivity. This phenomenon occurs when resistance to one antibiotic increases susceptibility to another antibiotic through shared or antagonistic mechanisms. In other words, the very mutations that allow bacteria to resist drug A make them more vulnerable to drug B. If we could map these relationships, which drugs create collateral sensitivity to which other drugs, we could potentially design treatment regimens that exploit these evolutionary trade-offs. We might treat with drug A, select for resistant bacteria that are now hypersensitive to drug B, then switch to drug B and eliminate those resistant strains. By cycling between drugs in strategic sequences, we might prevent bacteria from achieving stable resistance to any of them. Research on Pseudomonas aeruginosa has revealed that the stability of collateral sensitivity over time depends on multiple factors. The molecular structure of the antibiotic bacteria evolved sensitivity to, the strength of the original evolutionary trade-off, how sensitive the bacteria became, the order in which drugs were administered, and whether initial resistance came at large fitness costs. These aren't simple, universal relationships but complex, context-dependent interactions. Evolutionary experiments demonstrate three main outcomes when bacteria encounter the second drug after evolving hypersensitivity. First, bacteria commonly failed to counter the hypersensitivity and went extinct. The trade-off was too severe, and no ESKAPE mutations were available. Second, hypersensitivity sometimes converted into multi-drug resistance. Bacteria found mutations that simultaneously reversed the collateral sensitivity and maintained or increased resistance to the original drug, escaping the trap. Third, resistance gains to the second drug occasionally caused resensitization to the previous drug, thereby maintaining the trade-off in a different direction, potentially allowing the cycle to continue. Importantly, the order of drug administration significantly affected evolutionary outcomes. Starting with drug A and then switching to drug B produced different results than starting with drug B and switching to drug A, even when the same two drugs were involved. This suggests that optimal cycling strategies would need to carefully specify not just which drugs to use, but in what sequence to deploy them. Different bacterial species also exhibit distinct collateral response patterns during antibiotic resistance development. What works as a collateral sensitivity strategy in Pseudomonas might not work in E. coli or Staphylococcus. The genetics and physiology underlying resistance mechanisms differ between species, creating different trade-off landscapes. This species specificity means collateral sensitivity strategies would need to be tailored to specific pathogens rather than applied universally. Despite these complexities, collateral sensitivity represents an intriguing possibility, exploiting evolution rather than simply fighting against it, using bacteria's own adaptive responses to create vulnerabilities that can be therapeutically exploited. Combined with rapid diagnostics that quickly identify pathogens and their resistance profiles, and with computational modeling that predicts which treatment sequences would exploit collateral sensitivities most effectively, this approach could potentially guide antibiotic selection in ways that prevent or reverse resistance rather than merely delaying it. As we consider these various strategies, traditional combination therapy requiring multiple simultaneous mutations, dual-targeting antibiotics built resistance barriers into single molecules, and collateral sensitivity exploiting evolutionary trade-offs, we see medicine attempting to think evolutionarily. Rather than simply developing new antibiotics and hoping resistance won't emerge, researchers are trying to design treatment strategies that account for bacterial evolution, that work with and against evolutionary principles to maintain effectiveness over time. Whether these approaches can succeed at scale, in the messy reality of clinical practice rather than the controlled environment of laboratory experiments, remains to be seen. But they represent our best current thinking about how to stay ahead of bacterial adaptation, how to preserve antibiotic effectiveness not just for this generation of patients but for future generations who will also need these precious drugs. The strategies we've explored, combination therapy, dual-targeting drugs, collateral sensitivity, represent promising approaches for the future. But the reality is that most antibiotic use today involves well-established drugs prescribed in conventional ways. The question becomes, how can we use these existing antibiotics more wisely, preserving their effectiveness while still treating the infections that require them? This is the domain of antimicrobial stewardship, a coordinated effort to optimize antibiotic use in ways that benefit individual patients while protecting the collective resource that antibiotics represent. Stewardship encompasses many practices, but they all share a common goal: ensuring that antibiotics are used only when necessary, that the right antibiotic is selected for each infection, that doses and durations are optimized for efficacy while minimizing unnecessary exposure, and that treatment is narrowed or stopped as soon as clinical circumstances allow. Let's explore how these principles play out in several important clinical contexts. We begin with surgical antibiotic prophylaxis, the practice of administering antibiotics before surgery to prevent postoperative infections. This represents one of the clearest demonstrations of antibiotics' protective value and one of the most carefully studied applications of antimicrobial stewardship principles. Surgery, by its very nature, creates opportunities for infection. Incisions breach the skin's protective barrier. Surgical instruments and implants can introduce bacteria. Blood clots and damaged tissue provide nutrients for bacterial growth. For certain procedures, particularly those involving prosthetic joints, cardiac implants, or entry into contaminated body sites, the consequences of postoperative infection can be devastating, sometimes requiring removal of implants, prolonged hospitalization, or multiple additional surgeries. The effectiveness of prophylactic antibiotics in this context has been clearly demonstrated. A landmark study examining total hip and knee replacement showed that preoperative antibiotic prophylaxis reduced the absolute risk of wound infection by more than 80% compared to patients receiving no prophylaxis. Similar benefits have been documented across many surgical procedures, making prophylaxis standard of care for most operations. But the details of administration matter tremendously. The goal is to have antibiotic concentration in tissues at its highest at the start and during surgery, present when bacterial contamination is most likely to occur. Research supports administration at least 30 minutes, but no greater than 60 minutes before the skin incision for most commonly used prophylactic antibiotics. This timing allows the drug to circulate through the bloodstream, penetrate into tissues at the surgical site, and reach therapeutic concentrations precisely when protection is needed. Some antibiotics require longer administration windows due to their infusion characteristics. Vancomycin and levofloxacin, for instance, must be infused slowly to avoid side effects, requiring administration to begin within 120 minutes before incision rather than the standard 60 minutes. But the principle remains the same: antibiotics present at the right concentration at the right time. The timing of prophylaxis has been validated through careful clinical study. An analysis of 2,847 patients undergoing clean or clean-contaminated surgical procedures found that those receiving prophylaxis within 2 hours before incision had a surgical site infection rate of just 0.6%. In stark contrast, patients receiving antibiotics more than 3 hours after incision experienced a twofold increase in infection risk. Timing antibiotics after the incision, after bacterial contamination has already occurred, provides far less protection than preventing bacterial establishment in the first place. Current guidelines recommend that prophylactic antibiotics end within 24 hours of surgery completion. There's no documented benefit to continuation beyond this time frame except in specific circumstances like cardiac surgery where evidence supports 48 hours of prophylaxis. Prolonging prophylaxis for days or weeks, once common practice, doesn't further reduce infection rates but does increase antibiotic exposure, disrupt the patient's microbiome, create opportunities for antibiotic-associated complications like Clostridioides difficile infection, and contribute to resistant selection. This careful delineation, antibiotics started just before surgery, present during the procedure, stopped within 24 hours, represents stewardship in action. Antibiotics are used precisely when and where they provide benefit, and stopped as soon as that benefit ends. The approach maximizes protection while minimizing unnecessary exposure. Beyond surgical prophylaxis, antimicrobial stewardship programs have been implemented across diverse healthcare settings to improve antibiotic prescribing more broadly. These programs employ various strategies: education to improve prescriber knowledge about appropriate antibiotic use, clinical guidelines and protocols that standardize treatment approaches, prospective audit and feedback where infectious disease specialists review antibiotic prescriptions and provide recommendations, formulary restrictions that limit access to certain broad-spectrum or last-resort antibiotics without approval, and diagnostic stewardship to ensure appropriate use of tests that guide antibiotic decisions. The effectiveness of these programs has been rigorously evaluated. A comprehensive systematic review and meta-analysis published in 2023 examined 52 studies involving more than 1.7 million patients. The results demonstrated that antimicrobial stewardship programs were associated with a 10% reduction in antibiotic prescriptions and a 28% reduction in antibiotic consumption overall. That 28% reduction represents substantial decreases in unnecessary antibiotic exposure across entire populations. The reduction in consumption was observed across all major antibiotic classes. Prescriptions for penicillins, beta-lactamase inhibitor combinations, macrolides, fluoroquinolones, cephalosporins, and carbapenems all decreased significantly. This wasn't just shifting use from one class to another, but genuine reduction across the board. In pediatric hospitals specifically, stewardship programs were associated with a 21% reduction in antibiotic consumption, particularly important given concerns about antibiotic effects on developing microbiomes in children. Beyond reducing consumption, studies have demonstrated that stewardship program patients were almost twice as likely to be cured of their infection and more than 80% less likely to experience treatment failure compared to patients receiving standard care without stewardship oversight. These outcomes challenge a common concern about stewardship, that reducing antibiotic use might compromise patient care by undertreating infections. Instead, the evidence suggests that more judicious use, right drug, right dose, right duration, actually improves outcomes by avoiding ineffective antibiotics, reducing side effects, and focusing treatment more precisely on the pathogen causing infection. One of the most important stewardship practices is antibiotic de-escalation, the discontinuation of one or more components of combination empirical therapy or the change from broad-spectrum to narrower-spectrum antimicrobials after determining susceptibility results. The logic is straightforward. When infection is first suspected and the causative pathogen unknown, physicians often prescribe broad-spectrum antibiotics or combinations that cover many possible bacteria. Once microbiological results identify the specific pathogen and its susceptibility pattern, therapy can be narrowed to target that organism more specifically. A large study of 1,812 patients with hospital-acquired pneumonia and ventilator-associated pneumonia directly compared de-escalation to maintaining broad-spectrum therapy. The results were striking. De-escalation resulted in similar rates of 30-day composite treatment failure compared to maintaining broad-spectrum therapy, 35% versus 33.8%, a difference that was not statistically significant. In other words, patients whose antibiotics were narrowed did just as well as those maintained on broad-spectrum coverage. This demonstrates the safety and non-inferiority of de-escalation for treatment efficacy. But patients receiving de-escalation experienced several significant benefits. They received fewer total antibiotic days, reducing their cumulative antibiotic exposure. They experienced a 44% reduction in Clostridioides difficile infections, 2.2% versus 3.8%, a substantial decrease in the serious antibiotic-associated complication that can cause severe diarrhea, colitis, and even death. Hospital length of stay was reduced by a median of 2 days, 20 days versus 22 days, a clinically meaningful difference that reduces healthcare costs and allows patients to return home sooner. The impact on antibiotic use was rapid and substantial. By day 3 of therapy, patients in the de-escalation group experienced a 65% reduction in anti-MRSA therapies and a 44% reduction in anti-Pseudomonas aeruginosa therapies compared to the broad-spectrum continuation group. This demonstrates that de-escalation doesn't just slightly trim antibiotic use but makes dramatic reductions in broad-spectrum coverage while maintaining equivalent clinical efficacy. Another large study involving nearly 37,000 patients with community-onset sepsis confirmed these findings. De-escalation from broad-spectrum antibiotics to narrower-spectrum agents was non-inferior to maintaining broad-spectrum treatment. Patients whose antibiotics were narrowed based on culture results had outcomes equivalent to those whose antibiotics were continued unchanged. This multi-center evidence, spanning diverse patient populations and clinical settings, provides robust support for de-escalation as both safe and beneficial. The mechanisms by which de-escalation improves outcomes beyond infection treatment are multiple. Narrower spectrum antibiotics cause less disruption to beneficial microbiota, particularly the gut microbiome that provides colonization resistance against opportunistic pathogens like C. difficile. Reduced antibiotic exposure means fewer opportunities for selection of resistant bacteria within the patient. Lower antibiotic costs benefit both patients and healthcare systems. And psychologically, successful de-escalation reinforces a culture of judicious antibiotic use rather than reflexive broad-spectrum prescribing. The challenge in implementing de-escalation is timing and communication. It requires obtaining appropriate cultures before starting antibiotics, ensuring rapid processing of those cultures by microbiology laboratories, communicating results promptly to treating physicians, and having systems in place to review culture results and adjust therapy. In busy healthcare settings where physicians care for many patients simultaneously, this review process can fall through the cracks. Successful stewardship programs often embed infectious disease pharmacists or physicians who systematically review cultures and make de-escalation recommendations, ensuring the opportunity isn't missed. Given the success of combination therapy in some contexts and the logic of using multiple antibiotics to prevent resistance, one might wonder whether systematic approaches to varying antibiotic use across patients or over time might slow resistance development at the population level. Two such strategies have been proposed and tested: antibiotic cycling and antibiotic mixing. Antibiotic cycling involves scheduled rotation of one class of antibiotics with one or more different classes exhibiting comparable spectra of activity over defined periods. For instance, a hospital might use fluoroquinolones for 2 months, then switch to cephalosporins for the next 2 months, then switch to carbapenems, then back to fluoroquinolones, with the cycle repeating continuously. The cycle must repeat to fulfill the definition. It's not simply changing antibiotics once, but establishing a regular rotation. Antibiotic mixing takes a less structured approach, with consecutive patients on a ward being treated with alternative antibiotic classes. Rather than everyone receiving the same drug during a time period, alternating patients receive different antibiotics, creating heterogeneity in antibiotic exposure across the patient population. The rationale for both strategies was that they would reduce the selective pressure for resistance to any single antibiotic class, theoretically slowing resistance development. These approaches have been particularly studied in intensive care units, where antibiotic use is intense, resistant bacteria are common, and new strategies are desperately needed. Despite decades of interest and numerous small studies suggesting potential benefits, large-scale rigorous evaluation has been disappointing. A multi-center European cluster randomized crossover study published in 2018 assigned 8 ICUs to either cycling or mixing interventions and measured resistance rates over time. The conclusion was unequivocal. Neither strategy significantly reduced antibiotic resistance rates compared to standard prescribing practices. Ecological theory helps explain this failure. Mathematical models predict that heterogeneous antibiotic use, exposing the bacterial population to diverse selection pressures, should slow resistance spread better than cycling. At the bacterial population scale, mixing imposes greater heterogeneity than cycling because different bacteria encounter different drugs simultaneously, rather than all bacteria encountering the same drug during each cycle period. But even mixing didn't demonstrate clinical benefit. More sophisticated modeling suggests that cycling is unlikely to reduce either evolution or spread of resistance, and may even hinder resistance control in most clinical scenarios. The problem is that resistance genes, once they emerge, can spread through horizontal transfer regardless of cycling patterns. Cycling changes which resistance genes are favored during each period, but it doesn't prevent the accumulation of multiple resistance genes in single bacterial cells or on mobile genetic elements. Over time, bacteria carrying resistance to all cycled antibiotics can emerge, and the cycling no longer provides any benefit. These negative findings for cycling and mixing don't mean that varying antibiotic use is useless. Rather, they suggest that simple rotating schedules or alternating prescriptions, without consideration of local resistance patterns, bacterial ecology, and specific mechanisms of resistance, are insufficient. More sophisticated approaches that account for collateral sensitivities, that use rapid diagnostics to guide individualized therapy, and that integrate stewardship principles to reduce overall use while optimizing individual prescriptions may prove more successful. The lesson from cycling and mixing studies is that there are no shortcuts to stewardship. We cannot outsmart resistance simply through clever scheduling of antibiotics while maintaining high overall use. The fundamental principle remains: use antibiotics judiciously, only when needed, at appropriate doses and durations, and narrow or stop them as soon as clinical circumstances allow. This requires attention to each individual patient's infection and treatment, rather than population-level interventions that treat all patients similarly regardless of their specific circumstances. As we consider surgical prophylaxis, stewardship programs, de-escalation, and the limitations of cycling and mixing, a coherent picture emerges. Effective antibiotic stewardship requires detailed attention to the particulars of each clinical situation, guided by evidence-based principles but individualized to the patient, the pathogen, and the setting. It requires systems and infrastructure, rapid diagnostics, clinical decision support, specialist consultation, audit and feedback that support physicians in making optimal decisions under time pressure and uncertainty. And it requires a culture shift in healthcare, from viewing antibiotics as harmless and always beneficial to recognizing them as precious shared resources that must be protected through careful, judicious use. The data clearly demonstrate that when we implement these principles systematically, we reduce antibiotic use substantially while maintaining or even improving patient outcomes. A win-win that benefits both individual patients and the collective fight against resistance. As we near the end of our journey through the world of antibiotics and resistance, we turn our attention to several topics that don't fit neatly into the grand narrative we've been tracing, but are nonetheless important for understanding how antibiotics function in clinical practice. These are the details that can make the difference between successful treatment and failure, between unnecessarily restricted options and full therapeutic flexibility, between antibiotics that remain effective for decades and those that succumb quickly to resistance. Let's explore these special considerations that every clinician encounters but that often surprise those outside of medicine. We begin with what may be one of the most consequential myths in modern medicine: the belief that allergies to penicillin are common, severe, and extend across the entire family of beta-lactam antibiotics. This belief, held by patients and sometimes reinforced by cautious prescribing practices, has created a situation where between 8 and 15% of the U.S. population reports having a penicillin allergy. This is an enormous proportion. Tens of millions of people who believe they cannot safely receive one of the most effective and widely used classes of antibiotics. But here's the remarkable truth. Up to 95% of people who report penicillin allergies do not actually have a true allergy when properly tested. Some may have experienced side effects that weren't allergic reactions at all. Gastrointestinal upset, for instance, which is common with antibiotics but not an allergy. Others may have had viral rashes as children that were mistakenly attributed to the antibiotic they were taking for an ear infection or other illness. Still others carry labels from mild reactions decades ago that have since resolved. Many drug allergies, particularly those not involving severe reactions like anaphylaxis, fade over time as the immune system loses its sensitivity. The clinical consequences of having a penicillin allergy label, whether accurate or not, are far from benign. Recent studies demonstrate that patients reporting penicillin allergy have 50% increased odds of surgical site infection compared to patients without reported allergy. This increased risk is entirely attributable to the use of alternative antibiotics rather than beta-lactams. When surgeons cannot use cephalosporins or other beta-lactams for surgical prophylaxis, they typically turn to alternatives like clindamycin or vancomycin. These alternatives, while effective antibiotics, are generally less effective than beta-lactams for preventing the specific organisms that most commonly cause surgical infections. The excess infections resulting from avoiding beta-lactams represent preventable complications: additional surgeries to clean infected wounds, prolonged hospitalizations, delayed healing, increased pain and suffering, and in some cases, loss of prosthetic joints or cardiac implants that become infected. All of this occurs because of allergy labels that, in the vast majority of cases, are inaccurate. The myth extends beyond simply having or not having a penicillin allergy to beliefs about cross-reactivity between different beta-lactam antibiotics. For decades, medical teaching held that cephalosporins carried up to 10% cross-reactivity with penicillins, meaning that 10% of people truly allergic to penicillin would also react to cephalosporins. This figure, taught to generations of medical students and embedded in clinical decision-making, shaped prescribing practices worldwide. If a patient reported penicillin allergy, cephalosporins were avoided as potentially dangerous. But this oft-quoted 10% figure is based on unreliable studies from the early 1970s, and there's a crucial detail that undermines their validity. The cephalosporins used in those studies, manufactured before 1980, contained trace amounts of penicillin as contaminants from the production process. The apparent cross-reactivity wasn't cross-reactivity at all. It was direct reaction to penicillin contamination in the cephalosporin preparations. Modern manufacturing has eliminated this contamination. Recent higher quality data demonstrate that actual cross-reactivity between penicillin and cephalosporins is close to zero. Moreover, what limited cross-reactivity does occur is not a class effect. Not all cephalosporins reacting simply because they're cephalosporins, but rather a reaction to antibiotics with similar side chains. The allergic response targets specific chemical structures attached to the core beta-lactam ring, not the ring itself. Three-dimensional chemical structure analyses reveal that while cephalosporins and penicillins share the beta-lactam ring at their core, the side chains attached to this core determine immunogenic properties. These side chains vary substantially among different antibiotics. Some cephalosporins have side chains quite different from penicillins and show essentially no cross-reactivity. Others have more similar structures and show limited cross-reactivity in patients with confirmed penicillin allergy. Research has demonstrated that among persons with confirmed IgE-mediated penicillin allergy, the type of allergy involving the antibodies that cause rapid allergic reactions, the level of cross-reactivity with third-generation cephalosporins is low. Studies involving 214 patients with history of delayed hypersensitivity to penicillin, a different type of allergic reaction mediated by T cells rather than IgE antibodies, found that cross-reactivity occurred primarily with antibiotics sharing similar side chains to penicillins. Recent evidence shows that patients with testing-verified penicillin allergy can safely receive many cephalosporins, particularly those with dissimilar side chains such as ceftriaxone, cefuroxime, and cefotaxime. All carbapenems, imipenem, meropenem, ertapenem, and doropenem, can be safely given to penicillin-allergic patients because their side chain structures differ sufficiently from penicillins. The exact risk of cross-reactivity between specific antibiotic pairs with similar side chains remains largely unknown and likely varies among pairs, but avoiding all beta-lactams in penicillin-allergic patients is unnecessarily cautious and deprives them of highly effective therapeutic options. The implication is profound. proper allergy testing and delabeling of false penicillin allergies could substantially improve antibiotic stewardship and patient outcomes. Studies show that 97 to 99% of patients with reported penicillin allergy have no true allergy when tested, and these patients can safely receive cefazolin and other beta-lactam antibiotics without adverse reactions. Systematic programs to test patients with reported penicillin allergies and remove inaccurate labels have successfully expanded treatment options for vast numbers of patients, allowing them to receive optimal antibiotics for their infections and surgical prophylaxis. The lesson here is one we've encountered repeatedly. Assumptions that seem reasonable and become embedded in medical practice can persist long after evidence has undermined them. Questioning those assumptions, testing them rigorously, and updating practice based on current evidence rather than historical teaching is essential for optimal patient care. Now let's turn to two specific antibiotics with interesting properties that make them particularly valuable in the current era of widespread resistance. The first is tigecycline, a relatively newer antibiotic that represents an important tool for treating multi-drug-resistant infections. Tigecycline is a semisynthetic antibiotic derived from minocycline, itself a tetracycline. The structural modification, addition of a glycyclamide group to the minocycline molecule at the 9 position, places tigecycline in a class called glycyl cyclines. This modification was specifically designed to overcome the fundamental molecular mechanisms of tetracycline resistance that we discussed earlier: ribosomal protection proteins and tetracycline-specific efflux pump acquisition. The altered structure allows tigecycline to maintain activity even in bacteria that have evolved standard tetracycline resistance. Like other tetracyclines, tigecycline inhibits bacterial protein synthesis by binding to the ribosome, specifically to the helical area designated H34 on the 30S ribosomal subunit. But tigecycline binds with approximately 5-fold higher affinity than older tetracyclines, gripping the ribosome more tightly and making it harder for ribosomal protection proteins to displace it. This enhanced binding prevents the elongation of peptide chains, stopping protein synthesis and halting bacterial growth. Due to its superior ability to inhibit methicillin-resistant Staphylococcus aureus, vancomycin-resistant enterococci, and carbapenem-resistant enterobacterals, the very organisms causing the most serious resistant infections, tigecycline remains a valuable therapeutic option for severe infections when other antibiotics have failed. It represents one of the few drugs retaining activity against bacteria that have accumulated multiple resistance mechanisms. However, tigecycline is not immune to resistance evolution. Over the last decade, resistance has been increasingly reported through several mechanisms. Overexpression of RND family efflux pumps, particularly the ACRAB-TOL-C system we've discussed, can expel tigecycline from cells. Mutations in ribosomal binding sites can reduce tigecycline's affinity for its target. Regulatory mutations that upregulate various resistance pathways can decrease susceptibility. The fact that resistance can emerge even to this carefully designed antibiotic underscores the adaptive capacity of bacteria. Given time and selection pressure, they find ways around virtually any antimicrobial we deploy. Our second special topic antibiotic is nitrofurantoin, which offers a fascinating contrast to tigecycline. Rather than being a newer drug designed to overcome resistance, nitrofurantoin has been used to treat urinary tract infections since the 1950s. More than 70 years of continuous clinical use. Yet remarkably, resistance to nitrofurantoin remains exceptionally low. In an era when resistance to many antibiotics has skyrocketed, nitrofurantoin maintains effectiveness against the vast majority of urinary pathogens. Current susceptibility data tell the story clearly. 99% of E. coli, the most common cause of urinary tract infections, remain sensitive to nitrofurantoin. For Klebsiella strains, 69% retain susceptibility, and for Enterobacter, 63%. Compare this to conventional UTI drugs like ciprofloxacin and trimethoprim sulfamethoxazole, where resistance rates in E. coli have reached 25 to 29%. For nitrofurantoin, resistance stands at just 2.3%, more than tenfold lower. What explains this remarkable durability? Several factors converge to make nitrofurantoin particularly robust against resistance evolution. First, the drug achieves high concentrations specifically at the urinary tract site of infection through targeted renal excretion. The kidneys filter nitrofurantoin from blood and concentrate it in urine, where it reaches levels far exceeding those in blood or other tissues. This targeted delivery means bacteria in the urinary tract face very high drug concentrations that are difficult to survive. Second, nitrofurantoin exhibits a multifaceted mechanism of action. Unlike antibiotics with single, specific targets, nitrofurantoin disrupts multiple bacterial processes including protein synthesis and DNA synthesis. It doesn't inhibit one enzyme or bind to one structure. It causes broader cellular damage through reactive intermediates generated when bacteria reduce the drug. This complexity means that resistance requires compensating for multiple disrupted processes simultaneously, a far more difficult evolutionary challenge than overcoming inhibition of a single target. Third, development of clinically significant resistance to nitrofurantoin requires multiple independent mutations. Bacteria must lose or modify two genes encoding nitroreductases, NFSA and NFSB, that activate the drug. Losing one gene reduces susceptibility somewhat, but not enough to confer clinical resistance. Only when both are lost or inactivated does high-level resistance emerge. The requirement for multiple mutations dramatically reduces the probability of resistance arising, as each mutation must occur independently and both must be present in the same cell. Fourth, these resistance mutations impose fitness costs in the absence of the drug. Bacteria that have lost nitroreductase activity grow more poorly in antibiotic-free environments compared to their susceptible counterparts. This means that even if resistant mutants arise during treatment, they face competitive disadvantages once treatment stops, potentially being outcompeted by susceptible bacteria that retain the fitness advantages of functional nitroreductases. Fifth, horizontally acquired resistance genes for nitrofurantoin are nearly absent. While resistance genes for many antibiotics spread readily on plasmids and transposons, nitrofurantoin resistance doesn't seem to have been successfully captured and mobilized by these genetic elements. Resistance remains primarily chromosomal, passed only from parent to daughter cell, which limits its spread compared to resistance that can transfer between bacteria through conjugation. These converging factors, targeted delivery, multifaceted mechanism, requirement for multiple mutations, fitness costs, and lack of mobile resistance genes, create what one might call a perfect storm for resistant stability. Nitrofurantoin's success serves as a blueprint for designing future antibiotics that might prove similarly durable. If we could engineer drugs with multiple targets, require multiple mutations for resistance, impose fitness costs on resistant bacteria, and prevent resistance genes from being captured by mobile genetic elements, we might develop a new generation of antibiotics that remain effective for many decades rather than becoming obsolete within years of introduction. The contrast between tigecycline, a sophisticated modern antibiotic designed to overcome resistance yet facing emerging resistance within its first 2 decades of use, and nitrofurantoin, a simple older antibiotic that has maintained effectiveness for 70 years, offers important lessons. Complexity and cleverness in drug design matter, but so do fundamental properties like multifaceted mechanisms, high local concentrations, and genetic barriers to resistance gene mobility. Sometimes the simplest approaches, maintained consistently and used judiciously for targeted applications, prove most durable. As we consider penicillin allergy myths that unnecessarily restrict treatment options, tigecycline as a modern solution to multi-drug resistance, and nitrofurantoin as an example of unexpected longevity, we're reminded that antibiotic therapy involves not just the drugs themselves but the knowledge, beliefs, and practices surrounding their use. Correcting misconceptions, understanding each drug's unique properties, and learning from both successes and failures in drug design all contribute to our ability to use antibiotics effectively and preserve their utility for the future. As our long journey through the history of antibiotics and the challenge of resistance draws to a close, let your mind settle on this thought. The story we've traced is not one of inevitable decline but of continuous adaptation, innovation, and hope. We've traveled from the early 20th century discoveries that transformed medicine, through the mechanisms by which life defends itself, to the global crisis we now face. But the story doesn't end with crisis. It continues forward, toward solutions that draw on human ingenuity, natural wisdom, and international cooperation. One of the most powerful tools we have for addressing antibiotic resistance is also one of the most elegant: preventing infections from occurring in the first place. Vaccines represent this approach in its purest form. If bacteria never establish infection, antibiotics are never needed, and opportunities for resistance to emerge and spread are eliminated entirely. The numbers tell a compelling story. A 2024 World Health Organization report analyzing vaccines against 24 pathogens found that these vaccines could reduce the number of antibiotics needed globally by 22%, equivalent to 2.5 billion doses annually. That's 2.5 billion fewer opportunities for bacteria to encounter antibiotics, to evolve resistance, to spread resistant genes. The impact of vaccines already in use has been documented clearly. If we achieved the Immunization Agenda 2030 target of 90% global vaccination coverage for pneumococcal vaccines alone, we could save 33 million antibiotic doses per year. The real-world effects have been even more dramatic than anticipated. 4 years after the introduction of the pneumococcal conjugate vaccine PCV7, there was an 84% decrease in multi-drug-resistant invasive pneumococcal disease. The bacteria didn't become more susceptible to antibiotics. Rather, the strains carrying multiple resistance genes were prevented from causing infections in the first place, and their prevalence in the population declined precipitously. The WHO report suggests that vaccines could have averted approximately 515,000 of the 5 million deaths associated with antimicrobial resistance in 2019 alone. More than 500,000 lives that might have been saved through prevention rather than treatment. This prevention-focused approach represents a fundamental shift in how we think about combating resistance. Rather than constantly developing new antibiotics to replace those rendered ineffective by resistance, a race we seem destined to lose, we can reduce the selection pressure driving resistance evolution by reducing the infections that require antibiotic treatment. It's not that vaccines eliminate the need for antibiotics entirely, but they shift the balance, buying time and preserving effectiveness for when antibiotics are truly needed. Beyond vaccines, researchers are exploring alternatives to traditional antibiotics, approaches that work through entirely different mechanisms and might sidestep resistance altogether. Among the most promising is bacteriophage therapy, the use of viruses that naturally infect and kill bacteria. This approach, which actually predates the antibiotic era, emerged at the beginning of the 20th century but was progressively replaced by antibiotics after World War II in most parts of the world. Now, As antibiotic resistance threatens to return us to a pre-antibiotic state, researchers are taking a fresh look at these ancient bacterial enemies. Bacteriophages, often simply called phages, are remarkably specific, each type targeting particular bacterial species or even particular strains. This specificity is simultaneously their greatest strength and their greatest challenge. A phage that kills Pseudomonas aeruginosa won't harm E. coli and won't affect the billions of beneficial bacteria in our gut microbiome. This precision means minimal collateral damage, a stark contrast to broad-spectrum antibiotics that decimate beneficial and harmful bacteria indiscriminately. The mechanism is elegantly simple. Phages attach to bacterial cells, inject their genome, and hijack the bacterial machinery to produce more phages rather than allowing the bacterium to reproduce. The infected bacterium becomes a factory for creating its own destroyers. Eventually the bacterial cell lysis bursts open, releasing dozens or hundreds of new phage particles that can infect neighboring bacteria, creating a cascade of infection that tracks and destroys the bacterial population. The advantages extend beyond specificity. Phages have a high therapeutic index. The ratio between toxic and effective doses is very favorable because phages specifically target bacteria without interacting with human cells. They can replicate in vivo, at the site of infection, meaning that even small doses can be effective as phages multiply wherever bacteria are present. And perhaps most intriguingly, research has shown that phage-antibiotic synergy can be particularly effective. Phages can sensitize bacteria to antibiotics, lowering the minimum inhibitory concentrations needed for bacterial killing. Some antibiotics, in turn, enhance phage production and burst size, creating a collaborative effect where combined therapy performs better than either approach alone. The challenges of phage therapy: identifying appropriate phages for each infection, producing them at pharmaceutical scale, navigating regulatory frameworks designed for chemical drugs rather than biological entities, managing the specificity that requires precise bacterial identification, are substantial but not insurmountable. As resistance makes conventional antibiotics less reliable, the investment in solving these challenges becomes increasingly justified. These targeted approaches, vaccines preventing infection, phages specifically killing pathogens, fit within a broader framework that has gained prominence in recent years: the One Health approach. This integrated strategy recognizes that antimicrobial resistance cannot be addressed through medical interventions alone because resistance respects no boundaries between human medicine, veterinary medicine, agriculture, and the environment. The One Health framework brings together professionals from diverse fields: physicians, veterinarians, environmental scientists, agricultural specialists, public health officials, policymakers, to implement coordinated strategies across all sectors where antibiotics are used and where resistant bacteria can emerge and spread. It acknowledges what we've explored throughout this journey: that resistance developing in livestock doesn't stay in livestock, that environmental contamination creates reservoirs feeding back into human and animal populations, that antibiotics used in agriculture select for resistance genes that can transfer to human pathogens. The approach encompasses the entire antimicrobial life cycle, from innovation through disposal. It promotes responsible antimicrobial use, restricting antibiotics strictly to treatment purposes, rarely for prophylaxis, and never as growth promoters in agriculture. It strengthens surveillance systems to detect emerging resistance patterns early. It supports research into alternatives like vaccines and phages. It improves awareness through education across all sectors. And it preserves antimicrobial efficacy by ensuring that when these precious drugs are used, they're used optimally. The One Health approach represents a maturation of our understanding. a recognition that antibiotic resistance is fundamentally a systems-level problem requiring systems-level solutions. No single intervention in any single sector will suffice. Success requires coordination across disciplines, sectors, and national boundaries, sustained over decades. Even as we implement these preventive and alternative approaches, the search for new antibiotics continues. In March 2025, researchers at McMaster University announced the discovery of Lariocidin, a compound produced by Pinna-Bacillus bacteria retrieved from a Hamilton backyard soil sample. What makes Lariocidin particularly exciting is that it represents the first new class of antibiotics to potentially reach the market in nearly 3 decades. This lasso peptide molecule has a completely novel mode of action, binding directly to bacterial protein synthesis machinery in a way that no existing antibiotic does. The discovery reminds us that soil remains one of our richest sources of antibiotics. Soil actinomycetes, the group of bacteria responsible for producing about 2/3 of all antibiotics currently used clinically, continue to harbor undiscovered compounds. Researchers are now focusing on rare actinomycetes, species that are more difficult to cultivate, that grow more slowly, that might be overlooked in standard screening but that may produce unusual compounds with unique mechanisms. By employing genomics-based approaches and allowing soil bacteria to grow for extended periods, approximately 1 year in some studies, scientists are revealing even slow-growing species that would otherwise be missed, uncovering unusual biology and chemistry that could lead to future drugs. The potential of nature's chemical diversity is vast, but exploring it requires patience, sophisticated techniques, and sustained investment. Each new antibiotic class discovered represents years or decades of research, from initial isolation through characterization, optimization, preclinical testing, and clinical trials. The timeline is long, but the payoff, a genuinely new mechanism of action, can provide therapeutic options for infections that resist all existing drugs. Complementing this exploration of natural diversity, artificial intelligence and machine learning are opening entirely new approaches to antibiotic discovery. In 2020, MIT researchers demonstrated the power of this approach by using a machine learning algorithm to identify halicin, a compound that killed many of the world's most problematic disease-causing bacteria, including some strains resistant to all known antibiotics. The computer model screened more than 100 million chemical compounds in a matter of days, a task that would have taken human researchers many lifetimes using traditional methods. The system was designed specifically to identify potential antibiotics that kill bacteria using different mechanisms than existing drugs. After training on 2,500 molecules, the model was tested on the Broad Institute's Drug Repurposing Hub containing about 6,000 compounds. It identified halicin, a compound originally investigated as a diabetes drug, as having strong antibacterial activity with a chemical structure unlike any existing antibiotic. The researchers then applied the model to the Zinc-15 database, screening more than 100 million molecules in just 3 days and identifying 23 candidates structurally dissimilar from existing antibiotics. In April 2025, researchers developed an even more sophisticated end-to-end AI-guided antibiotic discovery pipeline spanning target identification through compound realization. They systematically evaluated 6 leading 3D structure-aware generative models, reducing over 100,000 generated compounds to a focused, synthesizable set of candidates. This represents a fundamental shift in how drug discovery can be conducted, from serendipitous observation and educated guessing to systematic computational exploration of vast chemical spaces that would be impossible to navigate through traditional experimental chemistry alone. Yet even with these powerful new discovery tools, the antibiotic development pipeline faces serious challenges. As of December 2022, approximately 42 new antibiotics were in clinical development worldwide that could potentially treat serious bacterial infections. A number that sounds encouraging until we consider the high failure rate of drug development. Most compounds entering clinical trials never reach patients. At least 18 of 26 phase I candidates were targeted to treat gram-negative bacterial infections, showing enhanced presence of novel antibacterial pharmacophores in early-stage trials. But translating laboratory promise into clinical success remains extraordinarily difficult. The stark reality is that only 2 new classes of antibiotics have emerged in the past 3 decades. Oxazolidinones, represented by linezolid, and cyclic lipopeptides, represented by daptomycin. The antibacterial pipeline remains scarce due to the costs associated with development and licensure, the complexity of conducting clinical trials for infections that may be uncommon or heterogeneous, and regulatory bottlenecks that have historically required large, expensive trials to demonstrate efficacy. Recognizing these challenges, the FDA issued finalized guidance in June 2025 on developing new antibacterial therapies for patients with serious bacterial infections and limited treatment options. This guidance offers flexibility, including smaller or single clinical trials, flexible trial designs that can adapt based on accumulating data, and acceptance of robust preclinical data when human data is necessarily limited due to the rarity of certain infections. These regulatory adaptations acknowledge the market failure that has driven pharmaceutical companies away from antibiotic development, the economic reality that antibiotics are relatively low-profit drugs compared to medications for chronic conditions, and attempt to reduce development costs while maintaining safety and efficacy standards. As research, development, and regulatory frameworks evolve, the need for global coordination and surveillance has never been clearer. The World Health Organization's Global Antimicrobial Resistance and Use Surveillance System, GLASS, represents the world's first collaborative effort to standardize AMR surveillance worldwide. Launched on October 22, 2015, GLASS provides a framework for countries to collect, analyze, interpret, and share antimicrobial resistance data in standardized ways, enabling meaningful comparisons and trend analysis across borders. As of the end of 2022, 127 countries, territories, and areas participate in GLASS, an impressive level of global engagement that reflects widespread recognition of resistance as a shared challenge requiring shared solutions. The system has expanded from initial surveillance of resistance in bacteria causing common human infections to include surveillance of antimicrobial consumption, invasive fungal infections, and implementation of One Health surveillance models that integrate human, animal, and environmental data. The 2025 GLASS report presents a global analysis of antibiotic resistance prevalence and trends based on more than 23 million bacteriologically confirmed cases from 104 countries in 2023. It provides adjusted global and regional estimates for 93 infection type, pathogen, antibiotic combinations, and tracks resistance trends for 16 combinations between 2018 and 2023. This vast dataset allows researchers and policymakers to identify where resistance is increasing most rapidly, which pathogen drug combinations are most concerning, and where interventions might be targeted most effectively. Surveillance is not solution in itself, but it is foundation for action. Without knowing where resistance stands and how it's changing, efforts to combat it proceed blindly. GLASS provides the eyes we need to see the problem clearly, to measure whether interventions are working, and to detect emerging threats before they become global crises. Yet even as we build these global systems and develop new tools, a sobering reality persists. The world's poorest countries experience the greatest rates of infectious disease and the highest levels of antibiotic resistance, yet suffer the biggest gaps in access to appropriate treatments. In 2019, Approximately 1.27 million people died from drug-resistant infections, with the majority of deaths occurring in low- and middle-income countries. The paradox is cruel. Those facing the greatest need have the least access. Multiple barriers compound this inequity. Poor political will fails to prioritize infectious disease control. Weak health systems lack the infrastructure to deliver care effectively. Complex bureaucratic protocols slow or prevent drug approval and importation. National action plans on AMR, even when they exist on paper, suffer from poor implementation. Inadequate expertise in regulatory science means countries cannot efficiently evaluate and approve new treatments. Unfavorable macroeconomic policies and poor investment climates discourage pharmaceutical companies from entering markets where profits are uncertain. In rural primary care facilities in Sri Lanka, for example, only 57% of medicines on the WHO Essential Medicines list are available, despite more than 95% of these medicines being off-patent and theoretically affordable. The gap between what should be available and what actually reaches patients in underserved areas is vast. The WHO reports that in some countries, approximately 80% of total consumption of medically important antibiotics occurs in the animal sector rather than human medicine, a pattern reflecting both overuse in agriculture and underuse in healthcare. The majority of the world's annual 5.7 million antibiotic-treatable deaths occur in low- and middle-income countries, where mortality burden from treatable bacterial infections far exceeds the estimated 700,000 annual deaths from antibiotic-resistant infections. Put differently, in the world's poorest regions, more people die from lacking access to antibiotics than from encountering resistant bacteria. The problem is not just resistance but also insufficiency. Not enough antibiotics reaching the people who need them, while in wealthier nations, overuse drives resistance that spreads globally. Addressing this inequity requires international resource transfer, capacity building, infrastructure investment, and a fundamental rethinking of how we value and distribute antibiotics globally. It requires recognizing that antibiotic resistance, like climate change, is a global commons problem where actions in one country affect outcomes everywhere. Investment in improving antibiotic access in low-resource settings, in strengthening infection control to reduce disease transmission, in providing clean water and sanitation to prevent infections, and in building healthcare capacity to deliver care effectively. These aren't charitable acts but enlightened self-interest. Resistant bacteria emerging in any healthcare facility anywhere can spread through international travel and trade to become everyone's problem. As we reach the end of this long exploration, having traced antibiotics from their discovery through their mechanisms, through the bacterial defenses arrayed against them, through the economic and environmental dimensions of resistance, through clinical strategies and future possibilities, we return to where we began, but with deeper understanding. The crisis of antibiotic resistance is real, growing, and threatening to undermine the foundation of modern medicine. The projections of millions of deaths by mid-century are not alarmist exaggerations but sober assessments based on current trends. Yet within this sobering reality lie reasons for hope. We understand the mechanisms of resistance better than ever before. We have tools for surveillance, for stewardship, for prevention. We're discovering new antibiotics and developing alternatives. We're building international cooperation and beginning to address the inequities that leave billions vulnerable. We're learning to think evolutionarily, to design interventions that account for bacterial adaptation rather than assuming permanence. The battle against antibiotic resistance will not be won through a single breakthrough or a magic bullet. How fitting that the term Paul Ehrlich coined for his first synthetic antimicrobial describes what we still seek. It will be won, if it is won, through sustained, coordinated effort across decades, through judicious use of existing antibiotics, through investment in new drugs and alternatives, through prevention of infections that need no treatment, through equitable access ensuring everyone can benefit from these life-saving medicines, through surveillance that tracks the enemy's movements, and through the recognition that antibiotics are shared resources requiring collective stewardship. The story of antibiotics is the story of human ingenuity confronting microbial evolution. It's a story that began with observation of molds on petri dishes and systematic screening of soil samples. It's a story that saved hundreds of millions of lives and enabled medical procedures that would be unthinkable without reliable infection control. And it's a story that continues, not toward an ending but toward an ongoing negotiation between human intervention and bacterial adaptation, a negotiation whose terms we're only beginning to understand how to shape. As you drift toward sleep, perhaps you might reflect on the invisible world that surrounds us, the ancient organisms that have shaped life on Earth far longer than our species has existed, and the delicate balance we're learning to maintain with them. The bacteria will continue to evolve. That is certain. The question is whether we will evolve too, not biologically but in our thinking, our practices, our cooperation, fast enough to preserve the medical miracles that antibiotics represent. The answer depends on choices we make collectively, starting now and continuing through the uncertain decades ahead. Rest well, knowing that across the world, researchers continue their work, physicians tend to the sick, and the slow, patient effort to preserve antibiotics for future generations proceeds, one careful prescription at a time.